Yes, yes, it works. We started at 10, right? Oh. Dwayne, there's no internet. I don't know. Internet doesn't work. Yeah, I get in and it doesn't work.
Okay, you have to select accept the disclaimer.
What is it?
You have to accept the disclaimer. Yeah,
I know.
it should open. Okay, disconnect.
Hello, everybody. Thank you for a wonderful day yesterday. I especially want to thank Dwayne, who did the heroics on the audiovisual and who was
in the blind tests. Sometimes other methods perform better, but generally we do well. So I think this is reproducible. And the second question is, can this be applied to non-coding mutations? We're working very hard on making that happen, but right now, no. And this is one of the important limitations of the method. Yeah.
Thank you. Oh, so we have to go on time, so sorry about that. We'll, hopefully can catch up in person. So next speaker is Carlos Cruchage from WashU, and he's an actual recording talk we're going to listen to. Dwayne?
Hi, this is Carlos Cruchage from Washington University. Today I'm going to talk about how we leverage human multiomic data to understand the biology of ApoE and to identify additional proteins and pathways that are involved in ApoE-dependent pathology. So, as we know, Alzheimer's disease is a very complex and multifactorial disease in which many cell types, including neurons, oligodendrocytes, astrocytes, endothelial cells, are involved. And I think that in order to really understand how these cells interact and how proteins interact between them and how they move across cell types, it's important to leverage human multiomic data. In my lab, what we have been doing in the last 5 years is to generate this molecular profiling of human data that includes brain samples, CSF, plasma, as well as PSEs. We are generating proteomics, metabolomics, transcriptomics, and other omics data. From sporadic Alzheimer's disease, Mendelian forms of the disease, but also for individuals with specific risk variants. And that includes, for example, TREM2, or in this case, ApoE carriers. And the goal of using all these omic data is to really identify new biological processes that are involved in disease, as well as to create new prediction models and identify potential new targets for drug repositioning. And at this moment, we have generated proteomics and metabolomics methylation in thousands of samples, in both cross-sectional and in longitudinal approaches. Today, I'm going to focus more on the CSF proteomics that we have generated in our 3,000 samples. Most of the data has been generated from the NIGHT ADRC cohort. And if you want to know more about the data and some of the analysis that we have done with that, you can take a look to this paper that was published a few weeks ago, in which we describe the data, but also how we can leverage the data and where is all the data available. So in the first part of the talk, I want to focus on a proteogenomic approach in which we integrate protein levels with genetics to understand how ApoE is involved in Alzheimer's disease. And in this case, where we are going to show is the proteomic data that we generate in CSF using the SomaLogic platform. In this assay, in this platform, there are around 7,000 proteins, and one of those includes ApoE. And when we do that, we see a very strong signal in cis for ApoE, which corresponds to the ApoE itself. So it's a cis signal. Then we have integrated this genetic data with the GWAS for Alzheimer's disease, and this is the Manhattan plot for one of the latest GWAS. And we have used a new approach, which is called fusion or TWAS, that was originally developed for transcriptome data, but that can be applied for other types of omic data, including proteomics. And what we are doing here is we are integrating the cis-QTLs with the GWAS for Alzheimer's disease to genetically infer the protein levels of Alzheimer's of the protein levels in Alzheimer's disease case and controls. And when we do this, and we have done this for ApoE, but for every other protein that we tested, we see that ApoE is the top hit. And also what it indicates is that higher ApoE levels are associated with higher Alzheimer's disease risk. We have done this for other proteins, and I have mentioned for 7,000 proteins, and some of those include non-Alzheimer's disease genes like TREM2, CR1, SERPIN, ACE, or others, but the top hit is ApoE. As I mentioned, this is basically a genetically driven ApoE levels. So we are inferring the level of the ApoE levels. So as the proteomic data that we use for this analysis is derived from Alzheimer's disease case and controls, we are also able to determine if we also can see higher ApoE levels in Alzheimer's disease. Instead of just doing Alzheimer's disease case and controls, in this approach, what we did was to stratify our individuals based on the AT status. So we have AT negatives here and ApoE positive in the extreme. And what we are seeing is that, in fact, ApoE levels are higher in A positive T negatives compared with AT negative. And we have seen this in both in the WASU data set as well as in NACC. So we can see that higher ApoE levels are found in early stage of the disease, but we don't see difference when we compare AT negatives versus AT positive. This is very interesting because for other biomarkers, including, for example, p-tau, we have seen similar trajectories in which p-tau goes high and then goes down. And that also indicates that we perform pure biomarker discovery based on clinical diagnosis, we may be missing some signals depending on what status Alzheimer's disease cases are. So this is a very nice example of how genetic can identify potential biomarkers, and they are not going to be biases for disease status, because all these proteogenomic approaches are integrating the genetic data and not just clinical status. We also see that higher ApoE levels can be found in ApoE4 positive as well as ApoE4 negatives. And we also see this trend that this association can be seen specifically where we are looking at controls and early stages of the diseases, supporting that higher ApoE levels are associated with higher risk for disease. So as I mentioned, we have done this for more than 7,000 proteins. And here is a summary of all the findings. This is what is called a 2D plot, in which each dot represents a genome-wide signal. And they are sorted by the location of the protein, of the pQTL, of the genetic variant, and the protein. And this diagonal signal is what we call cis variants. And also we can see that there are some vertical signals, and these are highly pleiotropic regions. And as you can see, there is the strongest pleiotropic region in CSF is the ApoE region. In this analysis, we found that there are more than 400 proteins that are associated with ApoE2 and ApoE3. And these proteins are enriched in neurons. So they are basically neuronal proteins. And these proteins are also involved in apoptosis and long-term potentiation. The first time that we saw this, we thought that what we are seeing is a proxy for neuronal death due to Alzheimer's disease, because Alzheimer's disease are in risk for ApoE4. That's what we are seeing. In order to demonstrate whether that was the case, we performed analysis in cognitive normal individuals, and individuals that were cognitive normal are biomarker negative, AT negative. And we saw the same association. The same proteins are associated with ApoE even before any clinical symptoms or biomarker positivity, indicating that, in fact, ApoE, which is mainly expressed in astrocytes or oligodendrocytes, is interacting with neurons and modifying neuronal activity even before we see any kind of pathology for Alzheimer's disease. And this kind of cell-to-cell interaction are the kind of things that we can do with these genetic approaches. Just to conclude, we had found that higher ApoE in CSF is associated with higher risk for AD. And this association is independent of the ApoE genotype, and it is specifically significant in early stages of the diseases. So if we want to understand how ApoE modifies risk for Alzheimer's disease, we also need to be aware about disease states because the protein levels are dynamic, and sometimes they are not just linear, but have complex trajectories. ApoE genotype is associated with the levels of 100 proteins, and those proteins are mainly highly expressed in neurons. They are part of the apoptosis pathways, and we can see this interaction before any clinical symptoms and are independent of AD pathology. And also, if we found or the community found a genetic therapy or a treatment that is targeting ApoE, these proteins that we are finding could be useful to track target engagement or treatment tracking, because we know if we target ApoE, these proteins also could change. So this also opens the possibility of finding or using these proteins for this analysis. I just want to finalize thanking everybody in the lab, mainly Dan Wester that did all the QT analysis, and everybody that has involved in the data generation, analysis, and so on. Thanks very much. And if you have any question, please let me know.
Yes, he's online. Carlos?
Yeah, I'm here.
Great. So there are several questions here.
Okay.
Oh, maybe.
Carlos, Jeff Vance, University of Miami. Really interesting, Carlos, as always. One of the questions about ApoE4 is how early since we're all born with ApoE4, how early do we start to see changes that are significant? How early have you looked at individuals, and could this approach be useful in developing ideas about timing?
Yeah, that's a good question. So in this study, we have quite a broad age range going from 45 to 90 plus, and included individuals in the different disease states. So we have controls that don't progress to disease, some that progress, and then different disease status. So those analyses can be done, but we have not really checked at what specific time these proteins start changing. So as I mentioned in the slides, in some of the analysis, when we found all of these proteins associated with ApoE genotype, we did check whether those associations are found in cognitive normal biomarker negative individual, and we keep showing those associations. So we think some of these changes happens very, very early on the disease course, but we have not do a specific analysis about what is the specific timing.
Next question right here.
Hi, Carlos. It's Mike Rachis from Stanford. Beautiful data, as always. There's this lingering sort of question about what ApoE genotype does to protein levels. So in the plasma, I think it's fairly consistently shown that E4 has lower protein levels than E3, than E2. And in CSF and in brain, it's sort of bounced back and forth. So I saw, the ApoE locus on your PQTL GWAS was a powerful hit. So do you have a definitive sense for what, the E2 versus E3 versus E4 looks like in terms of protein levels now in the CSF?
Yeah, so we found that there are differences across the genotypes, but those changes are dynamic, right? So I think that makes complicated about whether they goes up or goes down. So we see that initially the protein's level, independent of genotype, goes up with disease, but then goes down. So that complicates a little more about what is the absolute change because of this dynamic.
Yeah, so I guess, sorry to interrupt, but what about just in healthy controls, or pre-disease? Is there a main effect of the genotype on CSF protein levels?
Okay, that's a good question. We have not taken controls, so but that's a good. We can take a look to that.
All right, next.
Gil Paolo from Denali Therapeutics. Great talk, Carlos. Your connection with the neuronal apoptosis is interesting. It just raised the issue for me as to a potential connection with something like a neurofilament light chain. I wonder whether you have looked at the potential association of the ApoE levels with the NFL.
So the correlation of ApoE protein levels versus NFL protein levels, or the ApoE genotype versus the NFL?
The former, yeah. Everything protein level.
We have done that. I think we have a paper that was published like 6 months, and we take a look to correlation of protein levels in CSF and plasma for AD relevant proteins, and that includes NFL, ApoE, neurogranin, and some others that are in the somalogic panel, but I don't remember at this moment what that correlation is. Yeah, we can. Yeah, I don't recall at this moment.
Okay, thank you.
Next, Ellison or Daniela?
Alison Goat, Sinai. Great talk, Carlos, as always. I was wondering with the last analysis you showed with the master regulator, and I think you said that it was ApoE genotype that was driving that. So have you incorporated ApoE levels as well in that analysis to see whether ApoE levels as well as genotype impact that? And secondly, it seems like there's a really strong signal at the ApoE locus for ApoE levels.
Yeah.
And so I wonder whether you would try to dissect that more. Have you looked in, I don't know how many African-American CSF samples you have, but are you able to look in other ancestries and see whether you can narrow down the region that is involved and maybe potentially identify SNPs that are strong regulators of ApoE levels in CSF?
Yeah, so there is several questions there. So the first one, when we did the master regulatory region or looking at the pleiotropy of the ApoE, we have not correct for ApoE itself. So we can do that. We should be able to include ApoE in the model and ApoE protein levels in the model and see what's happened. But we haven't done that. So that's an interesting analysis that can be explored. As you mentioned, there are several signaling Cs in the ApoE region, and one is the ApoE protein itself, and others Nectin-2 also is associated with ApoE protein levels and ApoE-C, I think. So the analysis in CSF was done in Europeans. We have around 200 African Americans in CSF that we could run those analyses. We didn't run any African American specific analysis in CSF because we didn't have that much power for doing full PQTL discovery, but looking at specific signal that, like in this one, we should be able to do that. We did that for the CSF TREM2, and we used the African Americans to narrow down the signal in the MS4A region. But we have not done it for the ApoE at this moment. So
thank you. There's one more question here, and then we have one more question cover on Zoom.
Yazong Huang from Glasgow Institute and UCSF. It's interesting to see in the CSF, these ApoE levels is highest in the early stage disease, then drop when the disease further progress. Do you know what's the underlying reason for that? Is it because the ApoE deposit in the brain like A-beta, or is just the expression level change with the disease?
Yeah, so I don't know the answers to that one. We know that for other biomarkers like p-tau also have similar behaviors that goes up and down, first up and then down. And the specific mechanisms is not clear. We think it's also related with neuronal death and neurodegeneration in terms of a protein being found in the CSF. but we, at least myself, I don't know what is the specific mechanism.
Thank you.
Martin, can you repeat the question?
Yes, online question from Agustin Ruiz-Laza. Are SomaScan or ApoE SomaMass robust and accurate enough to confidently rely on the observed CSF findings? So question about reproducibility, I guess.
Yeah, so there are several SomaMers in the SomaLogic platform for ApoE, and one is for ApoE in general, and there are others that SomaLogic call them ApoE 2 and ApoE 3 and ApoE 4. So we don't think that the genotype-specific aptamers are really specific because we don't see, for example, we see that the ApoE4 aptamer measure ApoE in ApoE2s. So we don't think the aptamers are genotype specific, but we think there are ApoE, they are measuring ApoE in general. And one of the reasons is because we have a very strong signal in ApoE in Cs. So we see that that ApoE variants modify ApoE protein levels, including ApoE 2 and 4. So in general, when we see that in these assays, in SomaLogic or Olin, when there is a C signal for the protein, that support that these aptamers are specific to the protein. So we think we are measuring ApoE, at least one of the ApoE proteoforms. You know, we know that ApoE can be lipidated, can be glycosylated, and many other modifications. So which specific ApoE proteoform we are measuring, it's not clear, and we know that, you know, different assays can measure different proteoforms. So, you know, this is one of the problems of studying ApoE.
Thank you, Carlos, for attending this Q&A session. Thanks. Next speaker is Sally Temple from Neural Stem Cell Institute. She's going to talk about ApoE4 impact on vasculature.
Okay, let's get rid of this. It won't. All right.
To do.
All right, thank you, Sally.
Thank you.
So next speaker is Brad Hyman from MGH, and he is gonna be on it's a pre-recorded session.
Hi, I'm Brad Hyman from Mass General Hospital in Boston. I'm sorry I can't be with you there today for what seems like it will be an extremely exciting and important conference.
Can you volume up, Dwayne?
My disclosures.
It's pre-recorded.
We all know that ApoE4 is an incredibly strong risk factor for Alzheimer's disease. In fact, the strongest known genetic risk factor for Alzheimer's. But what I'd emphasize today is the equally strong role that ApoE2, the rare allele, has as a protective factor for Alzheimer's disease. This was evident from the early work from Alan Rose's group and our group, and then highlighted in Eric Reiman's work with a large population in which it was clear that inheritance of even one allele of the ApoE2 was strikingly protective in terms of age of onset. We examined the large NACC database as well as our own internal databases and showed that ApoE2 is associated with both a milder clinical and a milder neuropathological phenotype of Alzheimer's disease. So in addition to an almost 20-year difference in relative age of onset, in fact, with 2/2s, it's so rare that one can't even calculate an age of onset. There are fewer plaques and fewer tangles. So associated with a lesser risk, slower clinical progression, suggesting perhaps some sense of slowing or changing of the underlying neurodegeneration of Alzheimer's disease, fewer plaques and fewer tangles. ApoE impacts numerous aspects of Alzheimer's pathophysiology. Some examples from our own work: E4 is associated with an increase in the number of plaques in Alzheimer's disease, and all ApoE, E2, E3, and E4 are associated with plaques themselves. E4 is also associated with increased synapse loss and with the presence of oligomeric A beta at the synapse. ApoE genotype appears to impact microglial set point, such that E4 leads to more activated microglia, and E2 leads to a less activated phenotype, even at baseline, even without Alzheimer's disease. And then this is, of course, exacerbated in the context of Alzheimer's. As I noted, ApoE genotype impacts the clinical rate of progression, even after taking into account its effects on tangles and plaques. And David Holtzman's recent work suggests that ApoE4 impacts tau-mediated neurodegeneration via a neuroinflammation-mediated mechanism. So in each instance, E4 has a more aggressive phenotype than E3, and importantly, E2 has a milder phenotype than E4 or E3. We analyzed human transcriptome and subsequently single nuclear RNA-seq data sets and suggest that the impact of ApoE4 and the relative protection afforded by E2 might be due, at least to some extent, to an effect on microglial activation. So how can we take advantage of this idea that ApoE2 is protective in some ways, even in the setting of E4? Well, ApoE is a soluble protein in both the blood and the spinal fluid, but unfortunately it does not cross the blood-brain barrier. So that all the ApoE that's made in the brain is made essentially by astrocytes and microglia and to a lesser extent other CNS cell types, but essentially none of it is made in the periphery. In order to get ApoE2 into the brain, therefore, one would have to produce it in the brain using a gene therapy approach, or find a different way to introduce the protein into the CNS. In collaboration with Bev Davidson, we introduced an idea for getting ApoE2 into the brain and overcoming the problem that since Alzheimer's disease is a brain-wide phenomena, that the ApoE2 would have to be introduced in a way that it could bathe the entire brain. And Bev Davidson had developed a method to inject AAV directly into the ventricle to transduce the ependyma, the lining of the ventricle, leading to secretion into the parenchyma and the CSF, therefore bathing the entire brain in the gene product. In the example that we used here, we introduced 3 different doses of AAV into the ventricles. They all transduced the ependyma and not the parenchyma. ApoE was secreted, and by our measurements, roughly 10% of the endogenous amounts of ApoE were made and bathed into the parenchyma by this method. We then introduced ApoE2 into a mouse that was homozygous for ApoE4 and also APPPS1 to generate plaques and the plaque-related phenotypes. So in this instance, we're adding a little bit of ApoE2 to a constant and continuous amount of ApoE4 to ask the question, would exogenous ApoE2 have a protective effect? 60 days after the injection of the ApoE2, the mice were sacrificed for histologic and biochemical analyses. Looking at thioflavin S staining, or for that matter, a beta immunostaining, we see a dose-dependent decrease in the number of plaques over just 2 months. When we perform biochemistry ELISA for either SDS-soluble or formic acid-soluble A beta, similarly, a dose-dependent decrease in the amount of A beta present is observed. Excitingly, we also found that the presence of ApoE2 prevents microglial activation near plaques even for the remaining plaques, so that there are fewer plaques, and the plaques that remain appear to be less toxic to the microenvironment. We created an average microglial response score on a 0 to 4 scale, looking morphologically at the level of activation of microglia. We subsequently did repeat experiments with the CLEC7A and other markers of microglial activation. And the story is the same. Again, a dose-dependent substantial decrease, as is illustrated by the photomicrographs on the left, of how activated microglia are in the vicinity of plaques. Again, this is dose-dependent and very strongly related to the amount of ApoE2 present. As we all know, around plaques, there's a halo of oligomeric A beta and an associated loss of synapses. We asked whether or not the presence of ApoE2 could restore either of those phenotypes, and indeed, there's less oligomeric A beta around the plaques, and there is a nearly complete restoration of synapses. So in summary, we find that exogenous expression of ApoE2 via a gene therapy approach, even in the setting of continuous ApoE4, leads to diminished plaques, decreased microglial activation, and restoration of synapse loss. That is, a protection against neurodegenerative phenotypes in both amyloid, neuroinflammation, and clear neurodegeneration. Thus, ApoE2 acts in a non-cell autonomous fashion, suggesting that therapeutic approaches that mimic E2 in the central nervous system would allow E2 to cross into the brain or produce E2 directly in the CNS may be of benefit. I'll close simply by thanking my collaborators, Bev Davidson and her lab at CHOP, Dave Holtzman and his group at Washington University, the investigators in my own lab at Mass General, with a special shout out to Rosie Jackson, who has now taken a faculty position at University of Dundee. Thank you very much.
Brad is not on the line, but what I want to do is, if you guys have questions, go to the mic, we're going to record them, and I'll send the questions to Brad, and he'll answer them, and we'll post them at our website, the database, and okay? So ask your questions online and here.
Joachim Herz, UT Southwestern, Dallas. My question is when you overexpress ApoE2 in the brain virally, have you looked at to what level the endogenous ApoE4 production is being suppressed? Is it suppressed at all, and to what extent?
Anastasia, Khvorova, UMass Chan Medical School. My question is that if we look at genetics data right now from patients, it's a very rare mutations with ApoE3 null in a context of ApoE4. It seems to be that this is delaying disease progression. We have a lot of data from mouse genetics demonstrating that knockout of ApoE positively impact amyloid plaque formation and tauopathy. What do you think is going on with your data where you show that overexpression of ApoE2 positively impacted the biology?
So Martin, is there an online question that?
Yes, we have an online question
Yes.
from Najal. Great talk. What is the amount of ApoE2 expressed by this AAV compared to the endogenous levels of ApoE4? Additionally, are the ApoE2 lipidated? And then we have another question from Elias Wisdom. Have you or do you have plans to look at the Christchurch variant on these effects compared to E2, E3, E4? And a question from Reynaldo Oria. We expect to have AD patients to have some level of disrupted blood-brain barrier. Would peripheral injection of ApoE2 be helpful in these cases to reach the brain and improve AD outcomes? Any experimental data available from your group?
Thank you. So it's a nice connection from Brett Hyman study, and the next speaker, Ronald Crystal from Weill Cornell Medical College, going to discuss about the clinical trial of ApoE2 gene therapy. So he could cover a lot of aspects of E2 effects.
There's a question with this. You hit the big button to go forward, and backwards is the one beneath it. And you can either use the mouse to point on the screen, or the laser pointer, which push and hold and point it at the screen so the folks online can see. Okay.
Thank you. So let me give you an overview of our status of our clinical trial and the basis of it. It was initially funded by the Alzheimer's Drug Discovery Foundation and then licensed to Lexeo Therapeutics, and now is running the program. So the concept, as Brad Hyman articulated in the prior talk, is the idea is to convert the ApoE4 homozygote brain to an ApoE2, ApoE4 heterozygote brain. Since, as we've heard in this sessions both yesterday and today, E4 works on multiple pathways. So addressing the underlying genetics by converting the ApoE4 homozygote brain to E2, E4 brain could correct many of the downstream biologic mechanisms. So that's the basic concept. And the strategy is to use an adeno-associated virus gene therapy vector coding for the human ApoE2 gene, and therefore convert the E4 brain to an E2, E4 brain. The vector that we're using is based on AAV-RH10, which is based on a non-human primate adeno-associated virus. It uses an active promoter called the CAG promoter that functions in all cell types, very, very active, and coding for the human ApoE2 coding sequence. The initial preclinical data, Steve Paul's group and our group collaborated. This is just data from that study that we did a few years ago. And these were in the APP1, PS1 treat4 mice, so human ApoE4 mice treated with intrahippocampus delivery of the vector that I just showed you, and the data on the y-axis is A beta 142 and a dose-dependent decrease. And there's multiple other parameters, very similar to kinds of things that Brad Hyman showed. One of the challenges in terms of gene therapy is delivering it to not a mouse brain, but to a human brain. And the field has evolved and now it started off with catheters directly into the brain. And then over the years, we and many others studied administration to the CSF. This is data with non-human primates with the same vector that I just showed you, following intracisternal administration. Panel A is following several months after administration. We're looking at the monkey from the top left, and the monkey brain has been chopped up into 1 centimeter cubes. The lowest levels are the yellow, the highest levels are the red and the brown, and the green is sort of in the middle. So as you can see, in terms of where the vector goes when administered intracisternally in the monkey, it's distributed throughout the brain. In panel B on the right, and the messenger RNA levels are expressed for ApoE2 throughout the brain. And most importantly, the ApoE2 protein is found throughout the brain. So that's the approach that's used in the humans. The phase 1 clinical trial, and I'm going to show you data from cohort 1. The study population is greater than 50-year-old ApoE4 homozygotes. When I went to the FDA originally with pre-IND meeting, I tried to argue to treat ApoE4 homozygotes before any disease, but the FDA asked that we start with individuals with mild cognitive impairment and moderate dementia. These individuals have the CSF biomarkers and amyloid PET scan consistent with Alzheimer's disease. The administration is primarily, most of the patients at C1, C2, not intrasternal, so just one vertebrae down. It's dose ranging, and with originally the FDA asked us to dose per milliliter CSF, and so that data I'll show you is based on that. More recently, since the CSF volume turns out to be quite similar among the individuals, we've stopped doing that. Assessment over 1 year with a 4-year follow-up as required by the FDA. The cohort I'm going to show you does not have any immunosuppression. Subsequent cohorts have prednisone immunosuppression for 8 weeks. Endpoints of primary safety and the secondary endpoints of CSF conversion of ApoE4 to ApoE2E4 in CSF, and also data with CSF biomarkers, which I'll show you. So this is how it's administered, and the radiologist inserts the needle, as you can see, just in front of the spine. Takes about 1 hour. The patient is moved in and out of the CAT scanner. It's an outpatient procedure, and since it's a one-time therapy, these individuals don't have to be treated again. The CSF volumes run from 313 to 490, so not a lot of difference, as I'm sure you all know. As the disease progresses, the CS volumes increase. The total dose were ranged 2 times 10 to the 13 gene copies, and administered CSF I showed you. There's been no serious adverse events. There's been a few transient headaches in 2 out of the 5 subjects related to the administration, and a very minor pleocytosis of lymphocytes in a couple of patients, and no other serious adverse events, nothing related to the vector per se. This is the data for E4, E2, and ApoE2, E4. One of the questions that was just asked was, will the therapy have an effect on E4? So this is human data in panel A. One of the patients dropped out, and one of the patients refused to have a 12-month CSF, so we have 3 individuals out to 12 months. As you can see, the ApoE4 bounces around a little bit over time, but is not significantly suppressed. ApoE2 is expressed, as we can see, and it's expressed out to 1 year. And we're seeing about 0.5 to 1% of ApoE2E4 at this lowest dose in the individuals, with some variability as well. But we're seeing persistent expression out to 12 months, which is the length of the study where the CSF is being assessed. The E2E4 is quantified by liquid chromatography mass spectroscopy, and there are multiple runs per sample, so the variance is less than 10%. Also interesting is the CSF biomarkers. Again, very small numbers, but interesting. Panel A is A beta 42, and the yellow is 1 year. Blue is the pre. This 3 individuals from 59, 64, and 60 age. This decrease of 24%, 33% stability. In terms of total tau, a decrease of 20%, 4%, 22% at 1 year, and p-tau, 9%, 14%, 16% decrease. So again, very small numbers. The data for the additional dose groups are being analyzed now. To avoid batch effects in terms of these biomarkers, we're running them all at the same time, and those are being analyzed, and all the data for the other dose groups should be presented at the clinical trial Alzheimer's disease meeting at the end of October. And so in summary, at the low dose, it's safe, with no serious adverse events. ApoE2 is expressed in CSF at least.
And then let's share your screen again, good sir.
What would we do without Zoom?
Oh, no, they say now it works. No, yes.
Yeah, we're good.
All right.
Thank
you. All right, sir.
Okay.
You may continue.
All right, so I'll redo this panel. So just to give you some idea of second generation strategies, these are all studies that have been completed in mice. One is ApoE2 Christchurch. We've added the Christchurch variant to the ApoE2 and studied that in human E4 amyloid mice and tau mice. The data shows that sort of equivalent adding the Christchurch to the amyloid mice, but a dramatic improvement in terms of the tau mice. So that's a second generation strategy that we might go into the clinic with. The other two parameters that we've looked at is it's a good idea, we think, in terms of adding E2, but as we've heard throughout this meeting, E4 is toxic. And so another strategy is to suppress E4. And we've used two approaches to that, both with adeno-associated virus directed. The first approach is anti-ApoE microRNA expression in the CNS of ApoE4 humanized mice, and it suppresses ApoE4 levels. So that's one strategy, and that could be added to the E2 or the E2 Christchurch. And finally, most recently, we've done some gene editing, converting ApoE4 to ApoE3 with AAV directed prime editing to convert E4 to ApoE3 in the CNS of the TREE4 mice. And so altogether, we're moving along in the clinic with the E2 vector and considering adding some of these other parameters as second generation strategies. Thank you.
Hi, this is Mike Rachis from Stanford. Thanks for that talk. Thanks for putting this remarkable trial together. I had one, question I'm not sure I quite understood. You showed a graph showing the amount of D2 versus D4, or the ratio maybe?
Yes.
And I think, is it true that less than 1% of what you were measuring in the CSF was E2?
At the low dose, that's correct.
Do you have a sense from the animal studies what you'd like to achieve? Because that seems pretty low, I guess, the question. But I don't know.
Of course, in the mouse, it's a small brain and it's easy, so you can easily do it in the mouse and get very high levels in the mouse. But also in the non-human primate, and it's dose dependent. So although I don't know, we don't have the data yet, it's being analyzed, but I would expect to see a dose dependency in the human as well.
So that was really beautiful talk. Thank you so much for the pioneering studies. It's a safety study primarily. Given that we know ApoE4 can be beneficial for AMD, that it may have a slight improvement also for glaucoma, so you can have the opposite effect of these different isoforms in the eye. Did the FDA ask you to do any visual testing on the patients in the study?
So the answer is no. We actually do have some data in Alzheimer's in the eye in terms of corneal nerves. That has nothing to do with the gene therapy. The vectors, so the question is a good question, and you're asking is about systemic toxicity. We know that
within the central nervous system, right?
Yeah. So it's when we administer these vectors to the central nervous system, we've done PET scanning with I-124 labeled vectors, and we know that 50 to 60% leaves the CSF, and the CSF is cleared constantly, and so we know it goes systemically, and we see the liver and other organs light up. However, in terms of the eye, when you give these vectors intravenously, systemically, it doesn't go to the eye. So I would think it's not going to be a problem. But that's an important point, and that ought to be added to the study.
Thank you.
Thank you.
Hi, still Yingchen from Washington University, and that's exactly what I want to ask. Because according to your approach,
can you talk a little?
Yes. So that's exactly what I want to ask. According to your approach, you probably have some viral leaking from the CSF to the liver. In this case, do you also observe any change of the ApoE expression in the peripheral that may impact this phenotype?
No, we haven't seen any hyperlipidemia. We know that when you administer these vectors intravenously, which is essentially what's happening when it leaks out in CSF, is that probably 60–70% of it goes to the liver. And some goes to the heart and a few other places as well. But we haven't seen any systemic effects so far, at least at the doses that have been studied so far.
Okay, thank you.
Hi, Ron. Dave Holtzman from WashU. A very nice talk. In terms of ultimately translating this to humans and getting enough expression of ApoE2 in the central nervous system or other variants, what are the gene therapy approaches that are being used to, let's say you need to get, I don't know, 50% the amount of E4 or the same amount of, I don't know what the result would be needed, but are there other ways to administer the gene therapy other than just in the CSF that might, or is the CSF going to be adequate based on other studies that have been done?
Yeah, so it's a good question. There's two basic approaches. One is go directly, actually three approaches. You can go use catheters and go directly into the brain, and we've done that in some hereditary disorders. Second strategy is CSF. One, the way you're doing it into the ventricle. Another strategy is the way we do it, which is cisterna magna or C1, C2. But the other approach, where there's a lot of interest and effort in the gene therapy field, is systemic administration to develop vectors that cross the blood-brain barrier. And the field is getting pretty good at doing it. And if you look at the non-human primate studies that have been published, there's pretty good coverage throughout the brain. However, the problem is that it's only a very small fraction. You have to use very high doses to achieve that. So the field is moving along, and eventually it may be that systemic approach will be the best. But I think right now, probably the best approach is to go where the money is directly to the CSF.
Yeah, Tom Muriel, Columbia University. So two questions follow up on the last slide. One was that ApoE4 to ApoE3 conversion. What sort of efficacy are you able to achieve, sort of percentage conversion The other question I had was just sort of on a personal level, sort of, I know you're very involved in gene therapy. Do you think that's accepted now for patients? What kind of things do you have to communicate to the patients who are receiving the gene therapy?
Yeah, the data in the mice is about 20% in terms of conversion of E4 to E3. So it's a mouse, but reasonable amounts. I think as with the prior question, we know that the E2, E4 heterozygotes do so much better. But what we don't know is what level of E2 we really need. So it's 5% maybe enough, or do we need 50%? And until the human studies are done, we're just not going to know that. Yeah. In terms of your last question.
Yeah, I'm just curious, sort of the current acceptance of gene therapy and what you feel like as far as that being a viable option and acceptance from the patients.
I think it's going to work eventually. There's a lot of interest in the gene therapy field, not only us, but many groups, in terms of CNS administration and using. I mean, the real advantage of gene therapy is you get one time and you get long-term expression. And the second advantage is you get local expression, and the brain is a good example of that. And so I think it's going to take time, and I think we have to work it out eventually in humans, but I think it'll eventually work.
Yeah. Hello, Sebastian Malves, NextCure. I have two questions. In which cell types AAV10 transduce? Is it neurons, is it microglia cells, or others? And do you have integration of the ApoE2 gene into those cells?
Yeah, so in regard to what cell types, at least in the non-human primate, the AAVrh10 vector tends to be much more neuron than glia. So there's that. We and others have modified the vectors to get more glia if that is an advantage or so. Your other question is in terms of integration and concerns about integration of the vector. In the mouse, there's no question that there's some integration, and there has been malignancy seen in the liver. It has never been seen in larger animals or in humans, and there's human data now with these AAV vectors out for 7-8 years. So it's something obviously of real interest in the field, and there's a lot of data that's ongoing. But as far as we can tell, at least now, is that integration in the genome in terms of the malignancy risk is a mouse phenomenon and not seen in larger animals and never seen in humans.
Thank you.
To date.
So we have one more question here.
Yeah, Chris Ramsden from the intramural program of NIA. Great talk, really enjoyed it. I just had a question in terms of, do you think it's feasible to do a simpler, like caudal injection and then put the patient in decline position or something like that for a couple hours rather than trying to go in C1, C2? And if so, maybe you could protect it a little bit so that it doesn't.
You mean to go lower down?
Yes, to go,
Yeah, so there's a lot of data in that in non-human primates. And we've also evaluated that in non-human primates using I-124 labeled viruses, and so we can do PET scanning. And so what people have done is administer lower down and did Trendelenburg, it works. But at least in our experience, it's not as effective as going up higher. And so that's why we're doing it up higher. But there's a lot of interest in that in the field.
Sounds great. And maybe protect it a little bit so it wouldn't, so it could actually make it all the way up. But thank you.
So we have several questions online. Martin?
Yes, we have a question. Given that ApoE and Christchurch may offer different mechanisms of protection, is there any basis to co-inject E2 and Christchurch constructs?
There's not, do you mean to use two different vectors?
I think that's what they're asking. Yes.
I mean, the way we've approached it is to add the Christchurch to the ApoE2 in the same genome, in the same expression cassette. In general, there's not been, there's a few studies where people have injected two vectors at the same time. I mean, doubling the dose is not very much in viral terms, and so you could do that. And for one of the problems, at least in terms of The conventional approach for editing now is that you need 2 vectors because you have to split the gene into the Cas9 gene. So there is some data with using 2 vectors, but for the Christchurch, it's been in one genome.
Great. And another question was whether the individual biomarker reduction, A-beta, phospho-tau, etc., correlates with the individual CSF ApoE expression. I guess.
The answer is in general, yes, but I think it's too early to say. I mean, we would expect that. There's a rough correlation, but I think we don't have enough data to be able to definitively say that.
Great. Another question was the final slide suggesting that AAV injection of the Christchurch construct is more efficacious than the ApoE2?
It's in mice, so with that caveat, but it's more effective in the tau mice than the E2 alone. And so the effect in the tau mice is greater with Christchurch than we see in the amyloid mice.
Maybe a more general question here. What is the immune response of patients to AAV?
So that's an important issue, and one of the areas in the field that has been a lot of interest is in terms of using immunosuppression. So we have data from a prior study that we've done in CON2, Batten disease children, where we've measured neutralizing antibodies in the systemic circulation as correlating neutralizing antibodies against the vector, but in the CSF there were none. So that's probably blocked by the blood-brain barrier, at least at the levels of the neutralizing antibodies. What the field is doing is moving more and more to using immunosuppression for a period of 8 to 14 weeks. There's studies using prednisone, as we're now using in this study, as immunosuppression, but there's rituximab and a variety of other studies at all. What is not known yet for the CNS is how effective this immunosuppression is in terms of the immunity that might be ongoing within the brain. It's just not known, and it's going to take a few years to sort that out. And so if you look at the studies that are being done in the CNS, there's a wide range of no immunosuppression, prednisone alone, or prednisone plus other drugs. And it's just, the field is just sorting it out now.
Okay, thank you. We are on time. We're going to have a coffee break until 10:10, and we'll be back and continue this session. Thank you. Oh, we're going to start. Hello, we're going to start. So the next speaker is Guojun Bu, Hong Kong University of Science and Technology, and he's going to talk about the potential therapeutic role of peripheral ApoE, and this is a recorded talk.
Hello, everyone. Sorry I cannot be there in person today, but really happy to have an opportunity to talk about our own ApoE work. With today's topic, I'm going to focus on peripheral ApoE, which I will tell you actually have an effect on brain function as well as some of the pathways related to Alzheimer's disease. So here are my disclosures. So by now you have heard a lot about ApoE common variant, E2, E3, E4, and these differ at the amino acid 112, 158. But there are actually also several ApoE rare variants, and probably the two protected variants are best known. And as you know, the Christchurch has been shown recently by Dave Holzman, Yadong Huang, and Li Gan that limit the tau pathology and tau propagation. And these are consistent with what's been observed in the one homozygous individual in human. We've been studying an ApoE variant. We call this ApoE Jacksonville variant. And our work and also the work from Michael Gresh's lab at Stanford has shown that this reduced Alzheimer's risk by about 50% comparable to ApoE2 effect. So mechanistically, we've shown that this ApoE variant reduce Alzheimer's risk by really reducing ApoE aggregation. And this particular variant sits at the C-terminal of ApoE structure with a valine to glutamate mutation is actually reduced aggregation. And you can see that normally ApoE produced by HK cells come out as a tetramer on cytokine chromatography, whereas the Jacksonville variant would come out as a monomer. And this has been shown by wild-type cross-linking as tetramer, Jacksonville variants as monomer upon cross-linking. So monomeric ApoE in Jacksonville variant also make this ApoE form a better acceptor for lipid, as reflected in this lipid efflux assay. You can see that cholesterol efflux is greater with the Jacksonville variant compared to ApoE3. So there are many different pathways by which ApoE4 increase Alzheimer's risk. And likely the most important pathway is by driving the early and more abundant amyloid. And this actually also trigger many of the downstream effects, as you know. But there are also many other pathways that may not be A-beta dependent. And some of them relate to vascular function, such as the blood-brain barrier leakage associated with E4, perhaps metabolism, and also tau-mediated neurodegeneration. So again, these are all likely gain of toxic function. So we need to think about targeting E4 by reducing the toxic effect. There are also likely effect of loss of function. And even though this might not be dominant under pathological condition, but we should appreciate that the loss of ApoE, in, for example, depositing amyloid is associated with reduction of functional ApoE to mediate lipid metabolism, microglial function, and some of this might relate to the synaptic function and injury repair. So another complication of ApoE is that ApoE expressed by multiple cell types, both in the brain and also in the periphery. So in the brain, primarily by astrocyte, but also activated microglia, some in stress neuron. In periphery, primarily expressed by liver hepatocyte, but also macrophage. And these are separated by the blood-brain barrier. So vascular itself expresses abundant ApoE as well. So peripheral ApoE is associated primarily with large lipoprotein particles such as beta-VLDL and also a subclass of HDL. E4 is actually also a risk factor for hypercholesterolemia and atherosclerosis. The concentration of ApoE in periphery measured in the plasma is about 10 times higher than the ApoE concentration in CSF. So it's lots of ApoE in the periphery. There are actually epidemiology evidence that peripheral ApoE level associated with Alzheimer's risk. In this particular study, Copenhagen City Heart Study, there are about 10,000 individuals have plasma ApoE data. And when they separate out these individuals with 4 different levels of ApoE from high to low, 4 different quartile. And you can see that the lower level of ApoE is actually associated with higher risk for Alzheimer's disease and also higher risk for old dementia. And this is also sort of dose-dependent effect. Again, lower level of ApoE associated with higher risk for AD and old dementia. So we have performed a study where we look at a cohort of E3, E4 heterozygous individual. This was established and monitored by Rick Caselli at Mayo Clinic. And when we measure the plasma ApoE, so in this heterozygous individual, most of the ApoE accounted for by E3 isoform, with E4 only account for a small portion. So again, E3 correlated very well with the total ApoE, but not so much for ApoE4. Also, when we look at the ratio of E4 to E3, we found that the higher relative ratio of E4 over E3 is associated with gray matter volume loss and reduced glucose metabolism. So higher ApoE4 in plasma is associated with worse outcome. So to functionally address this, we develop an animal model where we express E3, E4 in liver hepatocytes. So this is a conditional model driven by the albumin cree. These are validation, ApoE expressed in the liver, not in the brain. The ApoE populate in the plasma, rescue the hypercholesterolemia effect seen in the ApoE knockout mice. What we saw was that functionally, the peripheral E3 in these Cre-positive animal enhanced memory performance in these field conditioning tests, contextual acute, compared to the Cre-negative animal, whereas in the E4 Cre-positive animal, there's a reduction of functional performance. When we measure the synaptic plasticity by LTP, E3 enhance this, E4 inhibit this. So mechanistically, we found that E4 actually increased blood-brain barrier leakage. This is measured by 2-photon microscopy with dextrin, fluorescent dextrin. And you can see that the positive E4 mice have greater leakage over time, and there are also reduced blood flow. We performed vascular enriched single cell RNA sequencing, so you can see these different clusters of vascular cells. One important observation we had was that E4 is associated with reduced astrocytic end feed, including the gene expression, aquaporin 4 here, and also when we evaluate the protein level by immunostaining, you can see that there's a reduction of aquaporin 4 quantified here when E4 is expressed. We also look at the plasma protein network. This is a collaboration with Streaming Peng. You can see that if we express E3, we saw increased expression of several proteinase inhibitors, such as a metalloproteinase inhibitor, TIM3, also serine proteinase inhibitor, SERPIN E2. Whereas if we express E4, we saw a reduction of protein involved in lipid metabolism, but an increased expression of some of the protein involved in inflammation or complement cascade. Finally, we bred these animals to the amyloid background without ApoE in the brain. The A-beta deposit mostly diffuse, but you can see that E3 reduce A-beta deposition, whereas E4 increase this. So in summary, we really shown that ApoE not only have effect in the brain in an isoform-dependent manner, but also peripheral E4 impair brain function, likely by impairing the vascular function, and also impair lipid metabolism, as well as promoting inflammation. So just to acknowledge individual did the work, Jenny Liu and Nancy Zhao. These work are primarily performed at Mayo Clinic. We're continuing this work at the Hong Kong University of Science Technology, focusing on the specific mechanism, also the ApoE2 effect. Thank you.
I think Bu is online, so can ask question. So it's question here, Bu.
Yeah, please.
Hi, really great talk. This is Michael Hain from UPenn. I was wondering if you've characterized the peripheral macrophages in your E4 periphery treated mice, if any, if they think that's contributing to a lot of the inflammation seen on the vasculature. Yeah, thank you.
Yeah, we have not done that specifically, but that's a very good point. I think inflammation is definitely one of the pathway that's sort of happening. You know, we show the profile, proteomic profile, and many of those could be related to macrophage. Macrophages themselves also express a lot of ApoE. There are a lot of classical study that, you know, dating back to Glasgow people studying vascular disease. So I think, you know, probably the alternative way to look at peripheral ApoE is to express different ApoE isoform in macrophage, just to see how they behave. But that's a good point. We haven't profiled macrophage yet.
Thank you.
Go ahead.
Hi, Bu. It's Mike Gratis from Stanford. Nice to see you virtually. Really lovely study. I guess you might have covered this, and sorry if I missed it in the quick 10 minutes, but so E4 made by the liver is opening up the blood-brain barrier a little bit. Is it possible, or did you look, and maybe I missed it, that E4 is also getting into the brain itself, you know, by the sort of breakdown that it fosters? Does it also get access to the brain that way?
Right. Very good question, Michael. Good to hear from you. I don't see you, but I think actually the reviewer asked that question, you know, when our manuscript was.
It wasn't me, I promise.
It wasn't you. So, but we look at it in several different ways. So immunostaining, ELISA, Western blot. I would just say we do not find detectable ApoE, But that doesn't mean that there won't be a few molecules of ApoE in the brain. Do keep in mind that these conditional mice express E3, E4 in the Cre-positive mice from birth. So, they actually expose ApoE4, E3 versus the Cre-negative, which is equivalent to knockout over this whole period of time. We look at these animals, I think 6 or 8 months. But in the paper, we did look at the younger mice. They did not have much blood-brain barrier issue. I think at 2 or 3 months they're fine. So there's some kind of degeneration event in the vasculature. So we don't fully understand, whether ApoE4 itself doing this or something that ApoE4 triggering, such as inflammatory molecule doing this. But a lot to ask there.
So, Bu, there are several online questions. Martin, are you there?
Yes, I'm here. Great talk, Bu. Do you think plasma ApoE could cross the blood-brain barrier in the presence of disease, at least in local brain areas? And along the same lines, have you looked at T cell and other immune cell infiltration into the brain in mice that have ApoE4 liver expression?
Right. So as I was saying, that ApoE in plasma, you don't really find non-lipided ApoE, and so this is really just a common knowledge in the field. But if you look at ApoE on lipoprotein particle, those lipoprotein particles are quite large. They're like probably about hundreds of thousand Dalton or a million Dalton. And if we look at the data, fibronectin or some of the protein we look at, they could be large and some of them cross the blood-brain barrier, but ApoE particles are much larger. So I think, under disease condition, there could be some pathological form get to the brain, but I don't think it's a major pathway contributing how E4 is impacting the brain. And we haven't looked at the T cell infiltration yet.
Do you envision liver expressing ApoE3 and ApoE4 positive patients during liver transplantation to improve brain and cognitive outcomes after surgery?
Absolutely. And I know there are colleagues working on that. In fact, one of our earlier inspiration was a liver transplant patient where, I think the individual switched from E4 to E3. The peripheral ApoE phenotype changed to E3, but brain remained to be E4. But I think from a study like this, I think it's not just our own study anymore, but I think there are a lot of evidence pointing that peripheral ApoE can impact what's going on in the vasculature, and probably some of this directly or indirectly impact brain function.
Great, and I guess a question that you partially already answered with this, but it's do you think the best therapeutic approach will be ApoE targeting in both the CNS and the liver, as opposed to CNS alone?
I think so. I mean, I think if you, let's say, if you do your gene editing, I think, ideally, because in peripherally, E4 is also, as I was saying, strong risk for atherosclerosis. So, if you change E4 to E3, I'm not quite sure about E2 yet, because it does associate with some vascular issues. But if you just change E4 to E3, it's actually benefit peripheral as well. But it does have function in the periphery when we think about the knockdown or antibody therapy. So there are areas we need to be cautious in terms of alternative approaches.
Great. One last question. The question was, in the mice used in the study did not express ApoE in the brain. What happened if you overexpress ApoE isoforms in mice that express ApoE at the normal level?
Good question. We're doing that experiment, but in the paper we published, we did do another experiment, which is to inject plasma from E3 E4 mice into a wild-type older animal, and just try to see, as you know, Tony Weiss-Corey work show that young plasma in particular can improve brain function. But what we found is that the young plasma in this older wild-type mice can improve function, actually enhance blood-brain barrier integrity, but E4 does the opposite. So we have some of this evidence, but obviously the genetic experiment can be done in either the wild type, or you can do E4 and E3 background, or mix them up. This could also be done with viral mediated approach, inject either peripheral in the brain. But that's a very good question.
I think we cover all questions. Well, thank you for staying up late at night over there, and you know you can go to bed. I'll see you later. Thank you.
Bye.
Bye. Next speaker is Hein Zhou from the Ionis Pharmaceutical, and she's going to talk about the antisense oligonucleotides for Alzheimer's disease, a focus on. Okay, thank you. Thank you, Hein. So, next speaker, Anastasia Khvorova from University of Massachusetts, RNAi modification of ApoE, the illicit balance between the plunge clearance and glia activation. Clearance.
Yeah. Forwards, backwards, laser pointer. Just push and hold and point it at this screen so the folks on live can see it.
Okay.
Okay.
Here?
Mm-hmm.
Oh, wow, it works.
Yeah.
Fantastic. Sorry, never have seen anything like that. Yeah. Perfect. Thank you so much for giving me an opportunity to be at this wonderful meeting. I learned so much. So what my lab is working on is developing oligonucleotide chemistry to make drugs out of them. And we're shifting the camps, going from antisense to siRNAs, which utilize or hijack the natural mechanism, RNAi mechanism, to silence genes at demand. As of today, there are 6 approved drugs and with siRNAs, and the winning clinical strategy is conjugation of fully chemically modified modalities. In this particular case, the durability of the effect, and we're not talking rodents, we're talking humans, it's up to 12 months following a single sub-Q administration. So if you look at the drugs, which is currently in either approved or in late stages of clinical development, there are 4 classes. First one, where most of the approved drugs are, all approved drugs are, is GalNAc conjugates. And GalNAc is recognized by the glycoside receptors, and this is liver only, liver hepatocytes only. Then there is a lipophilic assays, which are both explored systemically and locally, and antibody or antibody fragments of peptide conjugate, and the concept of multivalency, which my lab contribute to significantly. What is super important to understand that the oligo need to be fully chemically stabilized, meaning it's combination of sugars, phosphatides, noble backbone xNA, it's vinyl phosphonate on the 5' end, and you have to have it to ensure the 6 to 12 months duration of effect in clinic. So a while ago, we have reported a development of multivalent fully chemically modified SI, which show robust distribution in the CNS. And honestly, we just got lucky. We discovered it by accident, and in the long term, it makes sense. When you're increasing the size, you are decreasing the rate of CSF clearance, and the bulk CSF flow is a primary mechanism of the distribution of the compounds in the CNS. And what we have seen beautiful distribution in mouse brain, and people usually remember the pink brains. You can see silencing below level of detection, which can last up to 6 months, and most importantly, in non-human primates, you can see distribution throughout the brain, including deep brain structure, showing 80–90% silencing in cognitive entertainment, which antisense cannot do. So now when you have this chemical scaffold, you can apply and virtually silence any single gene in the human genome. And Atalanta, as a company we co-founded, have put in progress in different stages of development. So what I would like to talk about it, and this is some published work and non-published work, and how we can approach it to silence ApoE. So first, you do the screen, you identify the functional SI, you introduce it by intraventricular injections. In a non-human primate, it's spinal infusion, and in humans as well. And the first thing what we have demonstrated that if you look at the brain, look at the level of protein knockdown. So when you run the Western, you cannot see the ApoE. So at least in the brain, it's silenced below level of detection, not 50%. And in this case, when we use the doses, we start to see some level of decrease in the liver, which is not a good thing. But if you look at the circulating cholesterol, it's not affected. And it would be profoundly affected if you go with the liver targeting compounds. So the next thing we did is that while we don't see impact on cholesterol, it's ApoE, and you want to silence it in CNS selectively and have no impact on the liver. In ideal case scenario, you actually can do it with chemical engineering very easily. And I have no time to go into detail, but if you do a combination of compounds where you silence ApoE in the brain and then you block systemic efficacy in liver, you virtually get full silencing in the brain and absolutely zero impact on systemic ApoE. So the take-home lesson, easily, we can silence it in CNS only and nowhere else, and the silencing can last for 6 to 12 months. So we then went to I'm just showing you data for one model, but we have, of course, tested it in several. And in this particular case, it's 5x FAD mice. You can see complete silencing of ApoE, below level of detection at 2 weeks, below level of detection at 2 months post-injection. And what's surprising for us to see is specifically after David Holtzman's paper with 50% knockdown, that at 2 months post-injection, we've virtually seen no plugs, or the plugs are reduced very profoundly by 80–90%. So we also have seen reduction in insoluble tau, which makes sense based on data publications. So in general, what you start to see with untreated brain, there's plaques, there's microglia around the plaques, and with silence of ApoE, the plaques are out, down intracellular soluble APP is not affected, but lysosomal APP is decreased around 50%. So if we start to, and right now there's a couple of labs have shown exactly the same thing, you can see that if you co-stain for ApoE, and in this case it's red, and amyloid plaques, you see this co-aggregation. And most likely this co-aggregation of ApoE with amyloid, this is what's protecting it from microglia. Because what we do see a reduction in astrocytosis by GFP, but we see a typical picture of microglia surrounding this plaque, So in this case, the red is microglia. And when we remove the plaque quantitatively, we don't see pretty much any plaques. You still see this slightly activation microglia, but they are not clustered. The take-home lesson is ApoE clusters with amyloid. You remove the ApoE, amyloids start to be accessible to microglia clearance, and you reduce astrocytosis, but you're not reducing the amount of activating microglia. So we did the RNA-seq analysis, and it was very, very interesting because what we have seen, and it was unexpected, that we have seen an activation of microglia activation signature. And there's a lot of informatic analysis in AD paper, so you can look it up. But what was interesting to see is that the degree of activation is really directly correlated with the amount of plaque cleared. So if you look when there's not a lot of plaque, there is almost no activation. When there is low to medium plaque burden, you can detect it, but it's minor. And when you have a high plaque burden, which was cleared, you have a significant activation of microglia. Okay, so we can silence ApoE. Silencing ApoE need to be relatively potent, clearly more than 50%, and we can eliminate the plaque from appearance. And we suspect that what is happening is that destruction of this tight cluster of ApoE and amyloid beta is what making it accessible for microglia-based clearance. But what we also have observed that in this microglia-based clearance, there's some degree of activation and inflammatory response. And one of the things we have been working on for a while, and this is JAK1, it's actually a compound which will be in clinic for alopecia areata in February, but it's a master regulator of inflammation of interferon response pathways in a context of the brain. And there's certainly, like, for example, JAK1 inhibitors have been explored for the treatment of MS. So what we did is, it's another chemical engineering paper. So the only thing you have to remember is that we have this bifunctional compound. So we were able to make one targeting ApoE and another one targeting JAK1. And for some model targets with those bifunctional compounds, you can target both. So if you target ApoE, we do see this activation of the microglia signature. And then if we co-target ApoE and JAK1, you absolutely calm it down. It's not there. But with that said, when we injected those in 5-XAD, silenced both targets, I don't have a data there because it's just fresh off the press, is that we actually still maintain this pretty good level of the plaque clearance. So with that, I would say that we believe, and maybe it's a controversial statement, that all ApoE is bad, and ApoE4 is worse than ApoE3 and worse than ApoE2, but all ApoE is negatively contributing for misfolded protein clearance pathways. So we kind of feel that as a therapeutic strategy, the best way to go is to silence it to a significant degree. We're not sure you will need to do it for extended periods of time. It's quite possible that you might need to go with ApoE holidays, and the impact of that can be long-lasting. And because the ApoE and lipids in general serve as a buffer, buffering like microglia interaction with other structures in the brain, maybe at the time of ApoE modulation, tuning down either with bifunctional compounds or with existing JAK1 compounds, drugs, inflammation, maybe not a super bad idea. With that, I would like to acknowledge a huge group of people I have an honor of working with. It's amazing chemists, biochemists, cell biologists, and many, many collaborators. And as any academic scientist who's not NIH, I spend 50% of my time writing grants. Thank you for your attention.
So while other people prepare to talk, I have a quick question. So you said that reducing ApoE, like the cells are actually, microglia are more open to clean up the amyloid. And if you desensitize them by activating JAK,
we decrease the JAK, and I think the interferon signaling is happening not on a primary microglia function, it's on a secondary microglia function. It's like with inflammatory response, when microglia start to clear a lot of compounds, it sends over-exaggerated immune signal saying there is a lot of crap to clear, please bring me more. And I think if you shut down the second phase, you still allow the clearance, but you're probably potentially avoiding this over-exaggerating immune response due to the fact that you remove this barrier, protective barrier between crop and trash, which accumulates outside the cells, and microglia ability to clear it.
I'm Inter from Washington University. I'm very happy to see you actually notice the JAK1 and also the interferon response in the ApoE knockdown situation. So some paper has been published from groups, including one paper from on the immunity. They actually show that the type 1 interferon the signaling is actually inducing the microglia response, and that leads to the damage for the neurons and the synapse, which leads to the cognitive decline, arguably. I wonder, in your case, when you actually knock down the ApoE, you see the elevation of the type 1 interferon response. Do you also see the side effects? When the microglia are cleaning the plaques, do they also eat up the synapse, this kind of thing?
I think what we need to do, we need to kind of work with David Holtzman, because we are chemical biologists. Yeah, we can run a model and we can power it well, and we can look at the basic things well. With our level of expertise, we haven't seen any. We actually have seen a vast improvement in the overall performance of the brain, because the damage coming from plaques accumulation itself is probably far surpass the damage from a minor. It's not a huge activation, it's detectable, but it's minor activation of interferon response. But probably with your lab methodology, you might be answering those questions better.
Yeah, thank you. But yeah, I'm not trying to do that advertisement, but that's my question.
It's a great question.
Gil DiPaolo from Denali Therapeutics. Very nice work. Still around the theme of cleaning the crap, as you said. Do you think that it is the act of removing a beta by microglia that stimulates the interferon pathway, or do you have a potential scenario whereby the loss of the reduction in the levels of ApoE in microglia, which we know express a lot normally, could be, contributing to that interferon response?
So we have, I kind of went through that relatively quickly. We actually, when we silence ApoE in wild-type mice, which has no plaques, we do see slight activation in the interferon response, indicating that, and it's kind of a well-known fact, ApoE binds lipids, and when you bind lipids, you're protecting, you're essentially buffering all the interactions, not only with microglia, but with cells in general. With that said, it contradicts David Holtzman group, not contradicts, but David Holtzman group genetic studies didn't see it. Yeah. So genetic, you knock out ApoE in wild-type mice, and they're completely fine. And I think what we're going on when we're generating data right now, I think it's a timing scenario. If you have a brain which had ApoE and everything balanced, and then you shut it down within 3 days, and then you look short term, you see this response. But when you wait 4 to 6 months and the silencing is still there, it's kind of all re-equilibrate. So, but in a context of a plaque clearance, when glia is chewing a lot of dirt, yes, they get overactivated.
Thank you.
Long Huang from Glasgow, UCSF. Very nice talk. Questions. You see very profound reductions of the plaques. Did you also measure this freeform or diffuse A-beta levels also dramatically decrease or not?
So what we do see is when we're looking at A-beta levels or APP, intracellular APP is not affected, is the same. Lysosomal A-beta aggregation, because you have the soluble intracellular A-beta, is decreased around 50–60%. And the extracellular plaques A-beta is decreased 80–90%. So how the intracellular lysosomal A-beta is affected, I think it's something related to the fact that when we have those cluttered clearance pathways, when you already have so much A-beta accumulated externally inside the cells, this preformed soluble have no way to go, and it's there. And when you have everything kind of nice and cleared, you have indirect impact on soluble A-beta as well. So full-length intracellular APP is not affected, but lysosomal APP pre-aggregates are.
Did you also look at this vasculature amyloid in this model, or?
We are not as sophisticated to do it. So roughly by histology, we did not. There's no amyloids in the treatment group, and in the vasculature associated as well, but we have not selectively compare just generally plaques removal versus in vasculature. It's a very interesting question. What we do know is that we did a parallel experiment when we just used GalNAcs, which only deliver to liver, and we silenced ApoE in the liver completely below level of detection, and this has zero impact on amount of plaques in the brain. So we got cholesterol over the roof, yeah, absolutely, but no impact on what is happening within the brain. The disclaimer is the durability of those studies is 4 months. So you are doing it for 4 months, and in 4 months with level of sophistication, which is just histology and RNA-seq and biomolecular biochemistry assay, systemic silencing of ApoE did nothing.
So there are online questions, Martin?
Question from Agustin Ruiz-Laza. There's a double-edged sword hypothesis regarding the initial benefit of reparative inflammation in the early stages of the disease, followed by the harmful effects of chronic inflammation at the later stages. In your opinion, when should suppressive treatments be administered to maximize benefits and minimize harm?
I think the earlier is the better, because the more plaque aggregation you have, the higher is going to be the burden and potentially overexcitement of the microglia with that. And it's quite possible that if you go early in the disease as a prevention, you wouldn't even need to get core modulation of interferon signaling. But it can be beneficial in a later in the disease, because it will allow you to clear plaques no matter what, even when they are preformed. But in this case, the care should be taken to probably modulate overactivation of microglia response. Honestly, and I will say something probably very controversial, but I would say that Alzheimer is, at least late stage Alzheimer with all the genetic predisposition, is an exit-derated model of aging. And I think similar to Lipitor, similar to Ozempic, it's quite possible that this is just accelerated model of aging, and a lot of us early on might benefit from ApoE clearance procedure, not to do it permanently, but just to help a brain for a short period of time, just to cleanse this stuffed misfolded protein pathways. And you don't necessarily need to do it permanently, you just do it periodically. And how quickly do you need, how often do you need to do it? This really depends on, you need to test it in clinic. But at this point, we are seeing so much resistance, at least in the context of big pharma, from an idea that silencing ApoE is a good idea. And I think hopefully, what I'm really hoping that maybe a white paper coming from this meeting can help to push the needle towards a concept that at least it should be evaluated clinically.
So Anastasia, there are some other questions online. If you can join Zoom and answer it, we can answer. If not, we will post it on the website. So we'll move to the next speaker, combinatorial from the Francesca Vitali, University of Arizona, combination therapy in NACC and ADNI Alzheimer's participants, impact of ApoE genotype and sex.
Oh, okay, thank you.
Hi, good morning, and thank you for being here. And in this presentation, I'm going to introduce combinatorial therapy in NACC and ADNI Alzheimer participants with special focus on ApoE genotype. Alzheimer's disease is perceived by a preclinical phase that can start 20 years prior to AD diagnosis, and multiple risk factors such as diabetes, hypertension, and high cholesterol can contribute and accelerate AD onset, especially if not well controlled. Therefore, we ask the question if therapeutics currently approved to treat Alzheimer's disease risk factor can delay or treat Alzheimer's disease. And to answer this question, we interrogated medical informatics datasets to assess if FDA-approved therapeutics for AD risk factors are associated with an elevated or a reduced risk of Alzheimer's disease. In our work from 2021, we developed a strategy that was called Target Risk AD Prevention Strategy, in which we mined all the current literature to
So they should confirm it's back. Great. Thank you.
Okay, thank you. So after having identified the Alzheimer's disease risk factor condition, we used drug databases to extract drugs approved for this risk factor, and we identified 629 drugs. Then we developed a system score to rank these drugs by mining again PubMed literature, this time for the drugs, and if there is an evidence of a reduced risk of Alzheimer's disease associated with these drugs. And this led us to identify 46 drugs that are highly relevant and confident. And these drugs include hormone therapies, lipid-lowering therapies, anti-inflammatories, and metabolic drugs. And we, after performed a pathway analysis of the targets of these drugs, and we can see how there are these drugs of different categories, share targets, but also have unique targets, suggesting a combination therapies is maybe more beneficial for Alzheimer's disease. But also what emerging is, for example, that drugs within the same class, such as the statin, have different targets, and therefore also a precision therapeutics may be beneficial for Alzheimer's, especially, for example, for specific ApoE genotypes. I think it's blocked. Right. Okay. Okay. And in another study currently in preparation, we use this at the UK Biobank data, and we wanted to assess what is the impact of the major AD risk factor by, in this case, also by sex and ApoE genotype. And we use Cox proportional hazard methods to assess what is if the age of onset of the risk factor is also associated with the age of onset of Alzheimer's disease. And in these figures, ApoE4s are shown in orange, while ApoE4 non-carriers are in blue, and we can see that ApoE4s, as expected, show an accelerated AD onset, but that is different across the AD risk factors. For example, that hypertension and obesity have a more accelerated AD onset, while depression, for example, less, and also that for some AD risk factor, ApoE differences are more evident, while for other, like depression, is less evident. We then ask, so what if the medication to treat this AD risk factor has an impact on Alzheimer's disease in Alzheimer's participants? And in this case, we used NACC dataset and ADNI dataset to assess the impact of diabetes, lipid lowering, antihypertensive, and anti-inflammatory therapeutics. We extracted a total of 7,653 patients from the NACC data and 411 from the ADNI, and these participants have to have at least 2 visits and records of medication. And we selected as outcome measure the cognitive score of the MMSE and the CDR-SB score. The results, so we had patient group. We had a patient group by if they were prescribed one medication, a combination of 2, 3, or 4 medications. And we can see here that AD participants for which they were prescribed at least one medication showed a delay in cognitive decline if compared to patients that were not treated. And specifically, there was a 47% delay in cognitive decline in MMSE and 33% delay in cognitive decline with CDR-SB. And we can see that the quadruplets, so these percent are associated with the quadruplets of therapeutics, which is the one that has more beneficial effect on cognition. And even looking at a 2-year score change from baseline, we can see that the quadruplet shows already also a delay in cognitive decline for the CDR-SB, and also when selecting patients that were matched for baseline CDR-SB score, at 2 years, the quadruplets still show a delay in cognitive decline. And these results were validated using the ADNI dataset that again shows that the quadruplet was associated with a delay in cognitive decline for the quadruplets if compared with the non-treated patients. We asked then if there were any ApoE genotype differences in CDR-SB score change. And here we can see with the dot lines, there are patients that are not treated. The orange are the ApoE4 carriers, the blue, the ApoE4 non-carriers. And here we can see there is a slight worse performance for the ApoE4 as expected. And we can see that when patients are treated with a monotherapy, which is here on the diagonal, ApoE4 still, for example, for the lipid lowering, still show a cognitive decline comparable to the non-treated. However, when these lipid lowering are combined with, for example, hypertensive, their performance increases, and with reaching the best again with multiple combination of therapeutics. And in fact, here we can see that at 2 years, the ApoE4s are performing significantly different from the ApoE4 non-carriers, and with the non-treated, but at 2 years, the treated AD participants don't show any more a significant difference between the ApoE4 and the ApoE4 non-carriers. suggesting that these therapeutics also can bring ApoE4 carriers more similar to the non-carriers. So overall, we showed how combinatorial therapies in AD participants are associated with a delay in cognitive decline in both the NACC data and the ADNI data. And at 2 years, this delay in CDR-SD decline is comparable, if not greater, to the approved amyloid antibody therapies, which show that 27% at 18 months and 22% at 8 weeks. And this was surprising for us because we were not expecting that AD participants treated for the greatest number of comorbidity were also associated with, were exhibiting also greater benefit compared to the one non-treated and not having conditions also. And the NACC trends should include that we have a longitudinal assessment by sex and ApoE genotype that is extended up to 10 years. And while ADNI had fewer participants, these are more deeply phenotyped, and in fact, we also see greater delay in cognitive decline. And future direction will include the study of specific therapeutics within the class. So if specific statins are associated with better performances, and also including genetics for sure, because NACC also has data from genetics and ADNI as well, and extend also the analysis including blood-based biomarker and neuroimaging. And with this, I would like to thank all the team, and especially for this work, Dr. Brinton and Raymond and Georgina that really participated in this study, and to Coco-Let here. Thank you.
Great. I have a quick question. Julia T. Service from Boston University. So what stage of this patient, like what, you characterize all these AD patients, and which stage of patient did you treat these all the different drugs?
So these are already diagnosed patients, and
you analyze at which stage they are, or the long term of a drug treated.
Well, we have about 10 years, and we also adjusted all the patients at baseline to be comparable, like at baseline cognitive score, and by age at recruitment to avoid.
So those two strategies, MMSE and the CTR.
Yes, they were about like 21, I think, the average, and 5 for the CDR-SD.
Dr. Marcora Mansana, so what confounders did you adjust for to move from correlation to hopefully prediction of an intervention effect?
Yes, it's we adjusted for score at baseline, so and age at recruitment. No.
Hello, hi, Sebastian Malaves, NextCure. With respect to the combinations, usually means the contribution of components. So how can you assess components involved here, and what components will you get to benefit?
The compliance of the medication?
No, the contribution of components to combination. So which agent of the combination is going to be the main driver of the benefit?
I see. So in this study, the patients are assigned exclusively to one group. Okay, so the diabetes alone are patients that are only in diabetes. So each class are not overlapping. So it's kind of already exclusive.
So there are a few questions online. Martin?
Yes, starting with a question from Carlotta Pereira-Serras. Do you see any significant differences in drug effects by sex for any drugs?
Yes, exactly. We don't see much difference. It's not significant. So we see a difference, like if we separated male and female are still significantly, the cognitive decline is still significant. But there is not a very higher proportion in difference in any of the therapeutics alone.
Okay, question from Jose Cavazos. Were you able to examine the social determinants of health and education levels of controls versus treated comorbidity patients in NACC or ADNI?
Yes, no, we did not include the level of education in this analysis.
Question from Elizabeth Ria, a very interesting work, Francesca. Is it possible the controls have untreated comorbidities that may be influencing the results?
Yes, we have two control groups, one which is all the non-treated AD participants, and one is all the non-treated AD participants without any of the condition that we selected. So, and for both is showing.
Okay. And last question from Agustin Ruiz-Laza. Is it possible that polymedicated individuals have a mixed or greater vascular burden and less neurodegenerative burden, which could explain the charts you presented? Mixed and vascular dementia tends to have a delayed onset and a different progression compared to more pure AD cases, which evolve faster. How do you suggest addressing this possibility in the analysis?
Yes, that's interesting. And in fact, is it possible that we include also extend the analysis by stratifying or by vascular dementia or other dementia and see if there is a difference in what we are seeing?
All right, thank you. I think we cover all the questions.
Thank you.
So next speaker is Eric Reiman from Banner Health. He's going to talk about using biomarker in the persons with a different ApoE variant to isoform the study, treatment, and prevention of AD.
Well, thank you very much. Today, I'd like to share some thoughts about the use of biological measurements, including scalable blood-based biomarkers, to help inform the role of ApoE in the understanding, treatment, and prevention of Alzheimer's disease. These are my financial disclosures, including my involvement in prevention trials and the development of blood-based biomarkers. Neuropathological and biomarker measurements can help underscore the impact of ApoE4 and ApoE2 allelic dose on Alzheimer's disease risk. As we sought to show in this study of neuropathologically and clinically characterized cases and controls from the ADGC. You're familiar with the common odds ratios relative to ApoE3 genotype in clinically characterized cases and controls. And Guojun Bu, our ADGC colleagues, and I were interested in seeing what impact ApoE genotypes would have on a person's risk if we eliminated those clinically characterized cases who were misdiagnosed, which is about 25% of individuals with mild to moderate Alzheimer's dementia, 37% of ApoE4 non-carriers. And if we eliminated those unimpaired controls who have preclinical Alzheimer's disease, and you can see this dramatic increase in the odds ratios, you can see indeed a 0.004 odds ratios for being an ApoE2 homozygote compared to an ApoE4 homozygote. And this idea of about a 99.6% lower odds ratio in that group hopefully will kindle some interest in the idea of developing ApoE modifying treatments. Another advantage of these biological studies is the opportunity, as shown in this study and in one of the studies that Brian Hyman showed, of showing the residual effects of ApoE genotypes on tau pathology even after controlling for amyloid pathology. While all that is great, neuropath studies are limited in their size and with some notable exceptions, especially limited in the study of individuals from underrepresented groups. So antemortem biomarkers have the chance to address these issues in a large number of studies, including those in underrepresented groups, and also in prospective cohort studies. They could further inform the impact of ApoE4 variants on the risk of Alzheimer's disease in many more people. We have heard about these lovely studies, the landmark study of Lindsay Farrer, and this wonderful study conducted by Michael Belloy and Mike Grecius and their colleagues that have looked at the differential impact of ApoE4 on individuals from different ethnic, racial, and ancestral groups. And while this study is very important, it does beg an important question. How much of this effect is related to a lower risk of ApoE4 on Alzheimer's disease itself? Or how much might it be affected by the possibility that there's a lower percentage of Alzheimer's disease in these clinically characterized individuals? Some of us have this belief that we may see a lower prevalence of Alzheimer's pathophysiology in these groups. And one way to address these issues is to leverage legacy blood biomarkers from legacy samples to help to inform the prevalence and proportion of Alzheimer's disease and non-Alzheimer's disease dementias, including those in these different groups. Further characterize the impact of ApoE4 and ApoE2 allelic dose on Alzheimer's disease risk in particular, and then use those biomarkers to clarify whether the relatively protective effects that we are likely to see are related to resistance to amyloid pathology or resilience to the development of some downstream biomarker effects. So one of the take-home messages I would like to suggest is collect blood when you're doing your studies and think about leveraging legacy blood samples now. Unlike imaging, those samples may already be available to address some important questions sooner than later. A question that has generated a lot of interest since Juan Fortier's recent article in the Nature Medicine is are ApoE4 homozygotes destined to develop Alzheimer's disease and ensuing cognitive impairment? I raise this question in part as somebody who works with a lot of cognitively unimpaired ApoE4 homozygotes and heterozygotes who are understandably worried about their risk. We have consistent studies over the last 30 years that biologically and clinically characterized individuals from retrospective case control studies, the risk of Alzheimer's biology, which is now how we define Alzheimer's disease, and ensuing cognitive impairment is extremely high in ApoE4 homozygotes. But we have conflicting findings from prospective cohort studies, the kind of studies that we actually use to inform individuals not about their risk relative to ApoE33, but their probabilistic risk of a lower chance of developing Alzheimer's in their lifetime. So we have studies that go back to the original work at Duke that show, for instance, in that study of clinically and neuropathologically characterized cases and controls, over 90% of the ApoE4 homozygotes met criteria for Alzheimer's dementia. We saw something similar in the larger study of neuropathologically characterized cases and controls from the AVGC, Neuropath cohort. I wanted to include this figure from Willemijn Jansen's lovely data set in 20,000 individuals with PET and or CSF measurements, restricting the data to the cognitively unimpaired individuals in that cohort over a lifetime to show just how high those numbers look like in ApoE4 homozygotes, quite high in ApoE4 3/4. I personally think of this as a continuous difference rather than a categorical difference in homozygotes versus non-carriers, but it doesn't seem to be solely related to the issue of overrepresentation of cases in retrospective case control studies. And then we have the lovely work from Juan and his colleagues in using biomarkers in a very large number of individuals. They suggested that 75% of ApoE4 homozygotes had a positive amyloid PET scan.
That is what we mean with PET. PET evidence of at least moderately frequent neuritic amyloid plaques, which is required for the diagnosis of Alzheimer's disease. And that nearly all of them had low CSF A beta 42 measurements by the age of 65. Low CSF A beta 42 measurements, not quite as good as A beta 42 to 40 ratios for looking at amyloid aggregate deposition can be found, but it doesn't mean they all have Alzheimer's yet. It's suggesting some form of amyloid plaque aggregates beginning, and what happens to them over time, a little less clear just yet. When we look at our own studies in cognitively unimpaired individuals, I wanted to give you a sense of what we found. And really a kind of a massive effort for a prevention trial in a large number of ApoE4 homozygotes that was discontinued due to the adverse effects of a BACE inhibitor. We recruited about 640 ApoE4 homozygotes who are cognitively unimpaired, 60 to 75 years of age, and more than 2,700 ApoE34 heterozygotes. This is a study sponsored by Novartis with support from NIH and philanthropy, and this data set is available to the public, as well as limited blood and CSF samples in 400 of those individuals. And in this study, 64% of these individuals, average age 65, had positive amyloid PET scans. So not all of them had an amyloid PET scan. We went back and looked at the CSF A beta 4 reductions, and it's pretty similar to what was reported in the Nature Medicine article. More work to do. We'd like to see the A beta 42-40 ratios. In an ongoing study that's now gone on for almost 30 years now of cognitively unimpaired, late middle-aged ApoE4 homozygotes, heterozygotes, and non-carriers, amyloid PET measurements we found over time in work led by Valentina Giese suggest that after the age of 70, amyloid plaque measurements start to go down in those individuals who remain unimpaired. In Lindsay's 1997 report, the odds ratio for Alzheimer's dementia in ApoE4 homozygotes also begins to go down after the age of 70, possibly due to the later onset in ApoE in people with other genotypes, but possibly because there may be a lower risk of developing Alzheimer's disease in homozygotes who remain unimpaired after that age for other reasons we would like to figure out. We need to know for sure, and the sample sizes here are too small to know for sure. In prospective cohort studies, we don't see the same level of risk. When we were designing the prevention trial and knew we needed to inform individuals of their risk, we asked Deborah Blacker and her colleagues to conduct an estimate of risk from prospective longitudinal studies, including population-based studies. And in this study of individuals in the age group for our prevention trials, it turned out that 30 to 55% of the homozygotes, 20 to 25% of heterozygotes, and 10 to 15% of non-carriers were at risk for developing MCI or dementia by age 85. And those are the numbers we use to share with people about their risk. You might ask, well, maybe you missed a bunch of individuals who were already progressed. So we went back last week to look at those individuals who were about 50 years old when they were enrolled in our study and followed over the next 20 years. So between 50 and 70, for all the people who are asking us about what their risk is of developing cognitive impairment or dementia, and then before we find a prevention therapy, we found that a third of them went on to, the homozygotes went on to have cognitive impairment, and only 1 out of 18, in small numbers, 1 out of 18 developed dementia during that time. So we'd like to suggest that when we inform people about their potential risk, it's less alarming that we hear from retrospective case control studies, and that there's promising work happening in the area of prevention in the meantime. But we need, in the meantime, the findings from larger, longer, prospective-based, population-based studies to clarify the risk of Alzheimer's disease biologically and ensuing MCI and dementia in unimpaired individuals with each genotype. We need to understand the risk in the presence or absence of biomarker evidence of Alzheimer's disease, age and sex, so that we can provide that information as people will want to have them, especially when prevention therapies become available. And we want to get this information sooner than otherwise possible by surveying what legacy blood samples are out there so we can get that information soon. We've heard a lot about ApoE Christchurch in the last couple of days, and I just wanted to mention the data from this study we used to look at what turned out to be a rare ApoE variant, 2 copies of the ApoE4 gene, and use their biomarkers to try to understand what the mechanism was for its protective effects. If this individual was overproducing amyloid for an extra 30 years, was it because somehow we reduced amyloid plaque deposition even by then, or was it due to some downstream effects in the presence of a lot of amyloid? And because she was producing so much amyloid over that time, she had the highest amyloid plaque burden in the cohort, and yet had relatively protective effects on tau tangle and neurodegenerative biomarkers, providing additional support for the idea that we hear from really the elegant experimental work that ApoE has effects on other elements of pathology. One of the joys of doing a study like this, including just that first serendipitous observation, is to capitalize, work with our colleagues and collaborators from other areas, and the work that Joseph Arboleta, Cal Rose, Francisco Vitali, and I have had the privilege of doing with Yidong Wong and Bob Bailey has been a joy. You've heard about ApoE4 gene silencing treatments, and I share this view that ApoE variants are likely to be a toxic gain of function, with 4 worse than 3, worse than 2, possibly worse than Christchurch, worse than nothing. Well, in addition to having these opportunities to have a dramatic therapeutic effect in individuals, as soon as we have those gene silencing treatments that we can study in p-tau positive ApoE4 homozygotes, and hopefully with some other downstream effects, and show that the biomarkers go in the right direction with a small number of individuals relatively soon, we'll have the most definitive evidence we can to support that idea, and I think it will support other elements. So we would love the makers of the most promising treatments to make those gene silencing treatments available as soon as possible, that we can put them to the test in the right way, and we think these theragnostic biomarkers will play a critically important role in this endeavor.
Sorry.
Oh, really? Oh, I'm sorry. Let me discuss this area. In January, we published a perspective piece suggesting that there was a fighting chance to find and support the first effective secondary prevention therapies in unimpaired individuals who are blood test positive to avert clinical onset of Alzheimer's, and primary prevention therapies potentially used as infrequently as once per year in unimpaired individuals, older adults, ApoE4 carriers, starting before the onset of symptoms. We are hopeful that studies like this will start and give us the chance to find these treatments in the next few years. And we hope that ApoE4 heterozygotes and homozygotes who are worried about their risk will take heart in the possibility, even though we're confident that Murphy had clinical trialists in mind when he developed this law. And we look forward to working with them in that endeavor. I'd like to thank my colleagues and our research participants and wonderful supporters. Thank you very much.
So, any questions? Oh.
Lindsay Farrer, Boston University. Eric of the Tour de Force, thank you very much. I want to just point out a couple things. First, there is a paper published in 1996 that has been lost in the literature from Rick Myers, who looked at ApoE4 related risks, ApoE genotype related risks in the Framingham heart study. So that was the first and one of the few prospective cohort studies of that magnitude. And the point that I want to make, and I think this might be consistent with what you mentioned, is that among ApoE4 homozygotes, by age 78, only half gain dementia. Now, if there's still a period to go, and the confidence intervals were big because of the number of cases, but then that relates to my second point that related to your proposition that in the E4 homozygotes who make it to an old age and don't develop cognitive impairment or dementia, what's the holy grail, or what are the protective factors? And I think a couple of them were mentioned by Glia in her presentation yesterday. She did a GWAS among E4 homozygotes, and if I recall, the genome-wide significant associated variants were protected. The minor, the effect allele was protected. So I'm curious, I guess now the question, I'm curious what your thoughts are and what you might be planning to do to ferret out the protective factors in those before folks who we've accumulated quite a number of them.
Well, thank you for asking. As you heard, Mike has been a leader in that effort, and we have an ongoing study of 300 cognitively unimpaired individuals with each of the 6 common ApoE genotypes, including half of individuals who are older adults between 70 and 90. We're just completing with incredibly deep phenotyping. The challenge with studies like that with incredibly deep phenotyping is the small sample size. So what we want to be able to use is complementary data sets from highly phenotyped cohorts like that and very large cohorts like the UK Biobank and All of Us research program, for instance, who are very poorly phenotyped by reference. In those poorly phenotyped groups, it would be just very helpful to leverage the legacy blood samples and have endophenotypes of Alzheimer's disease to be able to look at the range of these factors and to be able to get that. We've been working hard. We have Jessica Langbaum and our team oversees the Alzheimer's Prevention Registry and a related gene match program. So we have roughly 400,000 individuals who receive information about the latest updates in research and participate in studies, and a gene match program with 120,000 people characterized by ApoE genotype so that they can think about options for studies and we connect them. We've been working hard to find those older adult ApoE4 heterozygotes and homozygotes, given all the hope that you see from the prospective studies. But for some reason, it's still hard to find. We're still working on it.
CJ, also sorry, because we are a little late. So we're trying to finish on time today. All those questions, can you please communicate after this talk? Thank you. All right. The last speaker is Joachim from University of Texas Southwestern. He's going to talk about therapeutic correction of ApoE4-mediated endolysosomal dysfunction in Alzheimer's disease.
Well, thank you very much for holding out for our last talk today. I want to talk to you about a different type of therapy, which we are currently investigating in the lab, which we have shown is not only correcting ApoE4-mediated effects on late onset Alzheimer's disease in cellular models as well as in animal models, but which has the potential to correct also the the dysfunction which happens in all forms of Alzheimer's disease, and that is specifically the endolysosomal dysfunction, which is increasing during aging. Okay? Now, we all know, of course, the hallmarks of Alzheimer's disease, but what is always considered almost as an afterthought is that these hallmarks are not appearing in individuals which are young, but they take a long time to develop, and late onset Alzheimer's disease more than 60 years. So what we were really interested in was why does it take so long? What other factors are coming into play? And the clue from that to that arose from observations which were made originally by Ralph Nixon over now 25 years ago, almost 30 years ago, who showed that in Alzheimer's disease individuals, another hallmark which is manifesting itself in the cells is that the endosomes are enlarged. Now, this is not only the case in Alzheimer's disease patients with early onset forms of disease mutations in APP or presenilins, but also this happens also in the presence of apolipoprotein E4, shown here in a cellular model, where ApoE4 endosomes are enlarged and accumulate in the cells. So we were wondering why is this happening, and we began to investigate this effect in cell models and in animal models. And what we found was that ApoE4 is causing this effect by inducing a traffic jam on the level of the early endosome. So ApoE4 has the same affinity for receptors as ApoE3. Both are efficiently endocytosed, but whereas ApoE3, effectively a retroendocytosis, is released again into the medium of cultured cells, and the receptors recycle back to the plasma membrane in unimpeded fashion. In the presence of ApoE4, ApoE4 is retained for extended periods of time on the level of the early endosome, trapping receptors and other co-receptors in cargo with it. And only a fraction of the receptors and of ApoE recycles back to the plasma membrane, whereas the rest becomes degraded ultimately in the endosome or is being released back into the medium by lysosomal exocytosis. So, and the question is, how does ApoE do this? And a clue which we derived from looking at apolipoprotein E primary structure came when we looked at the isoelectric point of apolipoprotein E isoforms. Now, it turns out that ApoE2 has the most acidic isoelectric point, and ApoE4 has the most basic one. And what was striking to us was that the isoelectric point of ApoE4 almost precisely matches the pH at the level of the early endosome. And it is well known that at the isoelectric point, proteins tend to self-aggregate, self-associate, and lose solubility. And in fact, this property was essential for the purification of insulin in the 1920s. It was already well known that to purify proteins, one can titrate the pH to the isoelectric point of the protein at which proteins tend to come out of solution. This was instrumental for the purification of insulin, and without this property, we would not have had any type 1 diabetes therapy developed in the 1920s. So based on that observation, we hypothesized that the same thing or something in a mitigated fashion like this would also happen to ApoE4, which then meant that ApoE4 would remain associated with its receptors for extended periods of time in the early endosome, thus preventing the resolution of the endosome and the further trafficking and maturation of this compartment. If this hypothesis is true, we then further rationalized that we should be able to interfere with this by simply lowering the pH of the early endosome further, accelerating the acidification. Now, there are two ways how one could potentially do this. One would be by raising the activity of the proton pump, which pumps protons into the lumen of the endosome, and the other one might be by blocking the primary proton leak channel, which lets proton flow out of the endosome. Well, we chose the second approach, because we can do this pharmacologically with so-called sodium hydrogen exchanger inhibitors, which essentially block the leak and therefore relieve the proton pump and accelerate the acidification of the early endosome. And the effect of this approach has been published by us in the paper shown here at the bottom. I had to take the slides for this out in the interest of time. However, this was quite effective, and this approach was also used, a different approach was also used by another group in another study, which used the C. elegans as a model system for aging and as well for the decline of endolysosomal acidification during aging. And they used this in this worm to study the effect of increasing endolysosomal acidification to clear proteotoxic products, polyQ, in a polyQ model, to increase longevity of the worm as well as restoring normal motility to the animals. So increasing, and this was done by using an antagomer directed against the mir, which down-regulates the components of the proton pump in the worm during aging. So both approaches apparently work the same way. You can remove the toxic products from a polyQ protein by increasing proton pump activity, or one can remove the amyloid plaques by plugging the proton leak channel and therefore increasing acidification of the endolysosomal compartment. So what we propose is that Alzheimer's disease and the onset of Alzheimer's disease may be determined to a large extent by how quickly the endolysosomal pH gradient deteriorates during aging, which would be happening earlier in an early onset form of disease, whereas centenarians might actually be protected by efficiently continuing to acidify the endolysosomal compartment, which protects them from the development of aggregation diseases. Now, this raises a possibility for a therapeutic intervention in a patient who is progressing towards Alzheimer's disease, either caused by the effect of apolipoprotein E or binding to receptors and requiring a greater level of acidification in the compartment, in the endosomal compartment, or by weakening of the proton pump during aging. Both would have the same effect. There would be a reduced influx of protons into the endosome, which leads to the enhanced influx of sodium or potassium ions and resulting in the swelling, which was noted in the tissues of Alzheimer's disease patients. Now, we can antagonize this by therapeutically preventing this imbalance in the pH homeostasis by partially inhibiting the sodium hydrogen exchanger, which then relieves the stress on the proton pump and restores the normal size of the endolysosomal compartment, which then allows for normal trafficking of cargo throughout the cell, including to the lysosome, as well as allowing their recycling back to the plasma membrane. So in the end, a key to Alzheimer's prevention may lie in the therapeutic rebalancing of pH homeostasis at the level of the endolysosomal compartment, which can be done by simply supporting the ailing proton pump during aging by partially blocking the primary proton leak channel. And this may be a preventive strategy which we all may want to consider, and perhaps which you might want to consider in your experiments to see whether this approach might help you to explain some of the phenotypes which you all are also studying. Thank you very much.
Joachim, I have a quick question. Julia, TCW. Is that the microRNA is turning on that transcription factor for the pump biogenesis?
Right.
Is that mechanism?
Yeah, so I didn't have the time to explain the MIR model, the C. elegans model very well. So what that group did was they identified a microRNA which is down-regulating the components of the C. elegans VATPase, proton pump, and during aging, which results in an age-dependent reduction of endolysosomal acidification. And what this group then did was form an antagomer against this MIR, which then failed to repress the transcription factor, which is regulating the expression of the VATPase.
Thanks.
Hi, this is Yunchen from Washington University. It's a great talk, and your work actually inspires a lot when we study other things, Christchurch mutation as well. So I guess my question is there any evidence from the GWAS studies that can tell whether any gene associated with the change of pH frequency in the endosomal trafficking system that is involved in this? And particularly in the ApoE4 carriers, is there any protective variant that is discovered that is associated with this process?
Okay, so the protein which we have identified, which is regulating pH homeostasis on the level of the early endosome, is sodium hydrogen exchanger 6, which is an X chromosomal gene. So that's only one functional copy per cell. And so the easy way of usually studying this by looking at are they heterozygous, are they protected, is not possible in this way. And it's not only us, but also Mike Gracious has looked in detail at the genetics of NHE6. And I think Mike might be able to comment a little bit more on that. But from our conversations, I can conclude that there is no statistically significant evidence that there are variants in NHE6 which are protective against Alzheimer's disease. But the numbers are very low, and this is difficult in X chromosomal genes.
Thank you.
Gil DiPaolo from Denali. Great talk, Joachim. I had a question about some of the work that you have published but not talked about today regarding the protective effect of the NHE6 NHE inhibition or ablation in the context of an amyloid model. So you saw a beautiful clearance of amyloid. I wonder whether what are your thoughts on the actual mechanism for this? Because it's not easy to sort of relate the ApoE2 work in neurons to the amyloids, right? So I wonder whether a possible explanation relates to the role of Glia simply clearing A beta, and this beautiful work from Fred Maxfield, for instance, showing that the acidifying liposomes in Glia can sort of accelerate the elimination of fibrils. Could that be a scenario as well that you consider in that context?
Yeah, so I didn't go into all the experiments, obviously, for the in the interest of time here. But our hypothesis was based on the self-association of apolipoprotein E, and we initially thought that this approach would be useful to just relieve the traffic jam which is caused by apolipoprotein E4. But then we actually did an experiment in which we used just mouse ApoE as a control, not human ApoE4, and the effect on amyloid and preventing amyloid plaque appearance was exactly the same. So we scratched our head, where is that coming from? Yeah? And until Ralph Nixon came out with another very interesting paper, a series of interesting papers over the last 2 years, in which he showed that in all models of Alzheimer's disease, there is actually decreased endolysosomal acidification capacity, including the models which we had used. So that immediately then suggested to us, well, we would obviously correct that as well. And if that is actually happening physiologically in vivo through a mechanism which I think needs quite a bit more work, namely the role of the beta CTF in suppressing VATPase assembly, as Ralph has proposed, but for which we have also now done some quite strong support that at least part of that model here is correct. then the whole issue of how is Alzheimer's disease developing and progressing may actually come down to the impaired acidification of the compartment, which leads to a slowing down of endolysosomal transport and sorting mechanisms over time, which then leads to amyloid accumulation, defective tau degradation, exocytosis of the aggregated proteins and so forth, everything which we know about how Alzheimer's disease is progressing. So I think it has potential to explain this mechanism quite well. And the purpose of me talking to you today was that perhaps you all should consider that that mechanism might be useful in complaining the phenotypes that you're all studying.
Chris Ramsden from Intramural NIA. Really great talk. As another commenter said, your work really inspired me to start a line of inquiry looking at ApoE receptor 2. And we've actually found that ApoE receptor 2 expression in the human brain, now 64 brains, is very closely correlated with p-tau vulnerability. So laminar, cellular, in every respect. And we're finding now up to 8 components of this pathway are all elevated, both surrounding plaques and intraneuronal as well. And again, a lot of that work came from where your publications, even a couple decades back in this. So I appreciate it.
Thank you.
So there are a few questions online. Martin?
Yes, one question is, does modifying the acidification with reagents affect only the intracellular environment of neurons or both the intra and extracellular environments? If both are impacted, could this influence normal neuronal activity?
Yeah, so one thing which I also did not mention is that NACC6, because it's on the X chromosome, is a genetic disease. When NACC6 is defective, it causes a genetic disease called Christiansen syndrome. In Christiansen syndrome, essentially there's no NAG there, so there's hyperacidification of the early endosomal compartment. And that has severe developmental consequences because that results in the premature degradation and missorting of neurotrophin receptors, which interfere with the development of the CNS. So I want to stress, however, that this is not a contraindication against developing NAGE6 specific inhibitors, because during aging we do not face a hyperacidification of the compartment, but an underacidification of this compartment. Therefore, a partial inhibition of the primary proton leak channel, NAGE6, would be expected to be beneficial. So I hope that answers the question.
Great, and I don't know if you have time for one more quick question. In which cell type could the ApoE aggregation be most efficiently eliminated by the acidification in the lysosome?
So what we looked at is if this works the same in fibroblasts as well as in neuronal cells. This may also explain why a lower plasma ApoE level is correlating with increased Alzheimer's disease risk, because apolipoprotein E4, as I mentioned, is being taken up by these receptor systems, remains in the endosomal compartment for extended periods of time, and then a substantial fraction of it is diverted to lysosomal degradation. So that would lower the plasma apolipoprotein E levels. So the more the ApoE is prone to reside and gum up the early endosome, the lower the plasma ApoE level would end up to be. Okay? So yeah, it works basically in every cell, and that mechanism is universal, NAG6 in every cell.
So I think we finish. We covered all the questions. Thank you. So we are gonna have lunch from 12 until 1 p.m. and we'll close this session 4. And from 1 p.m. for 2 hours. We are going to cover the session 5 about the brainstorming. And thank you, Martin, and thanks for the online attendance.
So I'm going to start in just 1 minute and hand it over to Dave. But I just want you guys to know we're using Slido today, this afternoon, and Dwayne's going to put up a QR code for you to snap if you want to use that. I understand we may also have access through the Zoom to chat questions. So either way will probably work. Also, I want to let you guys know that people are taking the bus the shuttle back to the hotel. We'll catch it at 3 o'clock where you were left off. Have a good brainstorm.
Well, thanks so much, Marilyn. Before we get started with our brainstorming session, I did want to mention an interesting and important meeting related to ApoE that's going to take place on March 17th and 18th in Miami, which is an Alzheimer's Association meeting on ApoE and lipid biology, and many of the same topics and other ones will be discussed, and hopefully many of you can attend that meeting. It should be very, very good. So as we get started, there's sort of, we came up before the meeting with 4 general areas of things that we wanted to brainstorm about. And I think it's going to be really important that we get great participation from everybody here, because this is a great chance after the meeting has occurred so far to really think about not just what we've heard so far today, but what do we think the field really needs to do going forward. It seems now that there's a great enthusiasm for trying to take advantage in many ways of what we've learned about ApoE to do things like develop therapy. But in doing that, there's a number of questions we still need to address. So having said that, why don't we can start by going over some of the points we put out before, but we're not limited to that. So I can start by just asking the first question that we raised for this first part of the session, which has to do with raising or lowering ApoE variants from a therapeutic perspective. So I think the first question that we put out there to potentially discuss is how to determine the optimal amount to lower ApoE4 and the effects of various ApoE isoforms and variants on ApoE4 lowering. How do we deal with lowering of ApoE in the periphery? So I know we discussed some of this during some of the talks, but why don't maybe this is a good thing to start off on and get some comments about. I don't know if people would like to say anything.
Hello. So first, let me start by thanking, of course, the NIA for organizing the workshop.
I'm sorry. Everybody say their name and where they're from.
Yeah, Susan Abu Shakra. I'm the Chief Medical Officer at Alzion. We're developing an oral anti-amyloid agent, which we also think has an ApoE4 corrector action. So let me just first say, it's very exciting to see all the science come together and some of the clinical reviews. The data on comorbidities is amazing. Just one thing, we're very interested in CAA because obviously that's one of the biggest risk factors for ARIA when people treat with antibiotics. So in the CAA literature, there's some evidence that having ApoE2/4 genotype is actually not somewhat harmful. They tend to have more frequent hemorrhage, and when they have them, they're large, spontaneous, large hematomas can be fatal in 50% of the cases. So I want to bring that up. Another point related to ApoE2 is that, in AMD, right, age-related AMD, it's also a risk factor to have ApoE2, not much ApoE4. So as we think about switching and changing the correct mode of action, just something I'd like your thoughts about.
One thing is, and this is just procedural, the fourth big topic we're going to discuss has to do with CAA. So why don't we save your first point to discussing that at that point? But it's certainly reasonable to discuss, the issue that E2 appears to be a risk factor for for retinal disease, for macular degeneration. But although the risk of the risk factor effect is nothing nearly as strong as it is, for example, for ApoE4 for AD. So I don't know. So
yeah, we were just talking about it, and I don't know how strong the data is for 2. Jonathan right now, but it's off the subject a little bit. But.
Yeah, it's off the subject a little bit, but the data for 2 is a little bit, is not terribly strong. The data for 4 being protective is stronger. But the odds ratios are not like they are in Alzheimer's. It's like 1.8 or something like that for the protection for the 4. The 2, as you pointed out, it's like maybe 0.9 something. I don't know. It's a little bit.
Yeah. So for getting at this first question, maybe I'll say something about one aspect of the issue is how do we deal with lowering of ApoE in the periphery? And I know that some people in the audience know this older data from the literature, but there's a number of studies that have been done in over 20 to 30 years ago showing that you can lower ApoE outside the brain by as much as 80% before you start seeing any effect on plasma lipoprotein metabolism. So I think unless there's something that I'm missing from the older literature, you ought to be able to lower ApoE quite a bit in the periphery without getting at least a major lipoprotein effect. I don't know, Joachim, if you and Yadong, I know you probably know this literature well as well, the older literature about this.
Yeah, just add on this. Like Dave just said, if you look at mouse work, it's basically without a high-fat diet challenge, even 5% ApoE maintain the normal lipid metabolism, 5% in mice. But if you put on the high-fat diet, probably 10% will be enough. This is known that in mice. In humans, of course, we don't know, but heterozygotes knockout clearly no problem with it. But based on the metabolic conversion of humans, you show people, I would guess probably 80% like Dave said. If you keep 20% normal output, the lipid metabolism should be maybe 90% okay, but to be safe, I think.
Do you want to go on?
I just wanted to comment. We know that in mice, if we in mice, if you silence ApoE 50% in the liver, nothing happens. 80% in the liver, nothing happens. When you silence it 95, cholesterol is over the roof. The only 2 comments which I wanted to make is while in mice it's safe, all the studies have duration of effect of 4 months. So what will happen if you have suboptimal ApoE in a context of a human for 2-3 years? The relatively good news, if we're talking about oligonucleotide therapeutics for modulating CNS-specific ApoE, the durability of the effect is very different. Like in CNS, you're looking for 6 months at least. And in liver, even if you see this 50% partial knockdown at optimal dose, within 2 weeks, the effect disappears. So maybe because of pharmacodynamics of the effects is so different, it's not the question of sustained modulation in the liver, it's temporal modulation in the liver is a lower degree. And hopefully that should be well covered.
Joachim, did you have
a... following up on what Yadong said, but on a bit of a cautionary note, the 5% of ApoE, residual ApoE, which is sufficient, comes from mouse data, and mice have a very different lipid metabolism than humans have. So I don't think I can extrapolate that face value. But one thing which one might also want to consider is if it just comes down to cholesterol, always have PCSK9 inhibitors. It's used by the PCSK9 antisense, which gives you enormous amounts of leeway. So I think ApoE can easily be reduced by 50% versus possibly more.
Well, if we look at the loss of function data, the individuals, and we assume that
it's 50% expression, although I guess we don't know that for sure, then certainly there's no reason not to think it. 50% seems to be fine, for sure. I have but there's another question towards that is that's for ApoE4, but if you wanted to knock down ApoE totally, is it the same amount? I'm sorry, if you want to knock down. So there's been some discussion of whether or not you should knock down 2, 3, and 4. Yeah, it's the same amount, I guess. Be 20% or so, 50%. Does it matter? I suppose it's the same. Don't think that means really don't clear. Yeah.
I just wanted to relay a message of encouragement from Francis Collins, who is excusing himself for not being here in person, but he's encouraging us to be bold in this very section, and we shouldn't be we shouldn't get our lack of knowledge on ApoE get in the way of mapping out potential clinical interventions directed at AD. We can spend another decade deriving basic science details, and we should, but we don't have to wait for all of the answers before planning therapeutic trials, which I think the field is doing in part, but I thought it was an important message from Francis.
I don't know, there's a point that's on Slido that I did Sudhaspadri that brings up that ApoE increases risk and E increases risk, and E2 the size of hemorrhage and CAA, so both are bad, unlike the benefit of E2 and AD. I think that is true. The only thing is that the percentage of what I don't know off the top of my head is what the percentage of people who actually are E42 that get CAA in the first place. And so, and also if you lowered ApoE of any type, it depends a little bit on when you do it in relation to when the pathology is present as to what might happen. So I think there probably needs to be, in addition to going for therapeutic trials, we probably need a few more animal studies to really understand how to address this, because there are good models actually for CAA in animals that have not yet been studied as much in regard to this question. So I think that's something that could be done. One of the other points we brought up is how long would lowering of ApoE4 need to be continued to have a long-term effect on things like A-beta time, neurodegeneration, inflammation, et cetera. And when during the course of disease can one lower E4 to get a protective effect? So obviously there was some talks about this in the last session. I think if lowering ApoE4, let's say, was able to affect the amyloid pathology, and if it really lowered it down, we know that it takes 20 years to go in a human to go from no amyloid pathology to what you see at the beginning of Alzheimer's disease clinical phase. So if you really were able to lower that pathology down to very low levels, like what's seen with the anti-amyloid antibodies, maybe you wouldn't need to treat for a long time with the agent. But it really depends a little bit on what the effect of lowering ApoE has on some of the outcomes. If you need sustained anti-inflammatory effect, then maybe you need to do it continuously. It really, I think, depends on what you see. And I think, as Francis was urging us, I mean, it almost seems like we really do need to try some things in humans with the appropriate agents that really lower ApoE effectively in the brain. Yeah, I think it was Anastasia mentioned that it, might be able to give it every, few years or something like that. Rather than a continuous dose, just knock it down. I don't know. But I do think that at some point we're going to have to address treatment in 4/4 carriers early. And because that's really where because it's going to take time to know. No matter what we do, it's going to take time to know that it's really working, right? Whatever we do, and we're going to have to follow them. Am I wrong on that? I don't think. Yeah, I mean, I think a lot of people who think a lot about treating Alzheimer's disease really think the biggest effects, of course, are going to be in prevention. Right. And so that would mean starting very early. If primary prevention would probably mean you'd have to start in E44 individuals by age 40, because they start getting amyloid deposition, many of them soon after that. There's some.
Yeah, I mean, just to comment on that, I agree. I think it's, Sal Nuriel, Columbia. I think that's the ultimate goal would be prevention, right? To treat an ApoE44 carrier, especially early so they don't develop Alzheimer's. I was struck though, Ron Crystal was saying that, he wasn't able to do that with the ApoE2 AAV, from an FDA standpoint. They didn't want to do that trial prior to treating, patients. So I'm curious if that would be a hurdle. The other question I had real quick was, obviously most ApoE carriers, ApoE4 carriers are ApoE3/4. Is it possible to only lower ApoE4 in an ApoE3/4 carrier? And would the ApoE3 sort of, continue to do its job in its place, and if that's a good strategy?
It sounds like at least with ASOs, it's very difficult to do that. I don't know. And siRNAs it may be similarly difficult because the region that's the difference is very GC rich. It might be, I don't know, Anastasia, if you have any information.
So we spent one postdoc year on trying to make a SNP selective ApoE compound, and with some chemical engineering, you can make it. The EC50 values are at least 50-fold lower than EC50 values for non-selective compounds. And at least in our hands, in mouse models, which is exaggerated mouse models, if you are silencing ApoE only 50% after plaque appearance, it's just not enough. So from that perspective, silencing just ApoE4 in ApoE4 heterozygous might be insufficient, at least in the patients where there is already certain symptoms. And we all talk about systemic ApoE, like liver ApoE, and saying that 10% of liver ApoE is sufficient to maintain the function, correct? So in CNS, we don't have such a robust data, but I think some parallels can be drawn. At least from the CNS data, we know 50% in established disease you can detect some fancy Folksman beautiful assay phenotypes, but the plaques are still there. If you silence it's 90%, the plaques are gone. So I'm not even sure and the third consideration is that, again, I do agree with Eric that all ApoE is bad. It's just different degree of bad. And from that perspective, having a drug which can work with all ApoE might be beneficial. But yes, you can silence ApoE4 selectively, but this will require 50-fold higher dose, meaning the therapeutic index is going to be dramatically lower.
Dado, did you want to?
Yeah, I mean, thinking about how much to lower it, how early. I'm thinking more from a pragmatic perspective, because eventually a company or companies have to take this on, and thinking about, uncertain PD and, preventative trials starting at 40 years of age. And also if we think that ApoE action is mainly mediated by A-beta, I don't believe fully, but in part at least. But now that they have agents that remove A-beta, why have companies not already engaged with ApoE, and what are the odds that they will, given the, especially this, uncertainties about the pharmacodynamics and how early you need to go to actually see an effect, especially if you want to go before antibody to see an effect.
Well, it's a good question. I think the one issue is if lowering ApoE4 is really protective against not just amyloid, but if it's really protective against neurodegeneration, then you certainly wouldn't need in the trial to initially start in a primary prevention trial. You could start a trial in some, people that are very mildly impaired, when really the tau and neurodegeneration phase are just kicking in. So I think that's sort of it's an animal model. The data that was shown yesterday and today shows that at least in a model of tau-mediated degeneration, knocking down ApoE4 by 50% strongly suppressed neurodegeneration. So I would think it's, whether that would be seen in a human, I don't know, but it seems it's good enough data to try that in an early clinical AD.
No, I don't dispute the science. It's more about, from the perspective of a company to invest and why haven't they already done so even before, and maybe, I don't know, maybe Jill has inside the I'm just wondering what are the investment factors are rather than, on top of the science, of course.
Yeah. I think it's a great question. I think the short answer is that there are a lot of ongoing efforts, and we can see some of those efforts, presented at this meeting with the ASOs or siRNAs, AAV approaches as well. I think there's been multiple factors that have played out in the past decade. One is, if the anti-ApoE type of approaches, and those approaches have been considered, continue to be considered, but they've been considered in the context of anti-beta immunotherapy. And I think that the issue has been also to what extent, and Dave can comment on this as well, to what extent the anti-ApoE approaches sort of replicate what an anti-A-beta antibody does, and not knowing for a long, long time whether the anti-beta immunotherapies have any clinical efficacy. It was a bit of a risky endeavor too, and so that has been a factor. I believe now things may be reconsidered in light of some of the more promising results obtained with anti-immunotherapies. There are some safety aspects which I know Dave has been thinking a lot about related to ARIA, and whether anti-beta antibodies, can solve everything, as opposed to alternate approaches that tap into the amyloid aspects without causing that level of ARIA that we are concerned about, particularly for the E4 carriers. So, and I'll say one last thing. There's been also quite a bit of work done on the small molecule space. With I'm sure you followed the literature on structural correctors, which in theory, amazing. It's an amazing concept to sort of try and convert the structural anomalies of E4 into a better behaved E3 type of structure. So far, those efforts have not panned out, and I think that has caused a bit of a cold shower in the field generally. But I think that a lot of these things can now be revisited, and I think are being revisited in many different companies, maybe through alternate approaches or approaches that more directly assess that. So I don't know if Dave, you want to comment on the differentiation from the anti-beta and how this has sort of been a factor for you as well.
So I think in very MCI or very mild dementia due to Alzheimer's disease, excluding the side effect issue of anti-amyloid antibodies, because there are some effects there, it looks like, in the studies that have been done in slowing decline by, let's just say, about 30% over 18 months. But that means that there's 70% that is still left. And so in that stage of disease, you would think that if what you're doing is targeting mechanisms that are beyond simply removing amyloid, that things like lowering ApoE4, which probably has effects beyond just affecting amyloid, it seems to me that would be a great add-on to a treatment like that, or done by itself, depending on whether the person is a candidate for that or can't be a candidate because of side effects or whatever.
Yeah, I was wondering that as well. A dual approach. If that's is that not only scientifically viable, but also from a public and corporate standpoint, that might be a way to, because they do have a problem with ARIA, and so that whether or not, but it's a thought. Also, I was wondering. Well, go ahead. Sorry.
I was going to say to answer that question. I think that the
we're heading toward a situation, presumably, where the anti-beta immunotherapies and maybe some improved versions of those anti-beta immunotherapies that are safer become the standard of care. So I think it's legitimate for all the companies doing work in that space to think about not only differentiation, but also what does our treatment add in the context of an anti-beta immunotherapy. So this is being considered for immune types of therapies on top of whatever anti-beta antibodies do. So I think it is entirely possible that we might be heading toward a situation where multiple drugs would be administered. They could be 2 large molecules, 1 large molecule, 1 small molecule, etc. But there's no anything is possible, I think, at that point. Yeah.
I mean, in most diseases, there's like different types of cancer. I mean, many combinations of drugs would be. So it wouldn't be surprising that you would need to do something like that.
We have about 6 minutes left in this section for this part.
Great.
Just a quick comment for how long. I personally think we should keep longer treatment, because if you stop in the middle, when ApoE come back, you can start the process again. So how long? I think if you look at the paradigms or not ApoE deficient humans are 40 years, 50 years age. In general, their brains don't have major problems now based on the published data, right? So it can go long enough. Peripherally, back to the key question, the problem on the hyperlipidemia, I mentioned one thing. Actually, mouse, humans, they rely on ApoE. In mouse, if you look at knockout mice, the hyperlipidemia higher than LDL receptor knockout. So in mice, because there's an ApoE48, maybe 1 P48, so you require a lot of ApoE in order to clear those remnant lines. That's why you knockout so severe hyperlipidemia in mice. In humans, actually type 2 hyperlipidemia, because FH with LDL receptor deficient, actually they have much higher lipid level cholesterol than ApoE deficient, because we have less P48 in our human body. So the fact of lowering ApoE impact, negative impact, might not be higher than
Go on.
Hey, Mike Rachis from Stanford. A couple things. I guess the first one, just to kind of reclaim my wet blanket approach from yesterday, is the last thing I'd want to do would be to muddy the waters, joint therapies like a novel ApoE therapy with these anti-amyloid. I think that's kind of going to be shooting us ourselves in the foot and not going to be clean most. You know, about 1 out of 4, maybe 1 out of 3 people on donanemab get ARIA. It's just be very messy. I also am not the least bit convinced that there's a meaningful clinical result in any cancer, antibody studies. So I don't want to sort of elevate them beyond the position. But the question of whether we should consider starting an anti-ApoE therapy even after amyloid plaque is developed is a big one, I think, right? If you know how hard it is to do primary prevention for adults. And I think what we really need, and we're starting to get, is encouraging NIA and NIH to fund these sorts of studies, is true longitudinal data on humans. What's been done so far to make the claim that ApoE has an effect not just on amyloid but on tau is to control for the amount of amyloid that somebody has cross section. But there's still a huge difference if you've had the amyloid in place for 15 years versus for 5 years. And now with plasma biomarkers that they're mentioning with amyloid PET, we can really say, okay, this person became amyloid positive 4 years ago, this person 11 years ago. Does E4 have an effect after that, controlling that on tau neurodegeneration? Because it's conceivable. Obviously, the animal models suggest that there are lots of non-amyloid effects, but we haven't firmed that up in humans.
Well, yeah, I think you make a great point in that we do have some really, at least in Alzheimer's disease, some great biomarkers for different aspects of pathology, whether it be amyloid, tau pathology now, neurodegeneration, inflammation. So all of those need to be integrated into whatever the trial is done using therapy. And it would be crazy not to do that.
Eric Reiman from Banner.
Is that okay? Very kind. So I agree with several of Mike's points, but not with all. There are ongoing secondary prevention trials of unimpaired individuals with biomarker evidence of Alzheimer's disease. The donanemab, that's a trial that we collaborate with Lilly on, and lecanemab ahead. The postulate is that in unimpaired individuals where amyloid plaques have been there for a shorter duration of time and lower level, will have a greater response to treatment, will have less ARIA, even in ApoE4 carriers. The hope is that there will be a relationship between the treatment's biomarker effects and the clinical benefits so that it might help to accelerate the further evaluation of a range of treatments. The results of some of the first studies may be as early as a couple of years from now. I think we just need to wait to see where we are in that space about benefits versus the risks while we continue to work on what we need is a more diverse portfolio of prevention therapy opportunities, including ApoE modifying treatments. Not too far off.
I think the AHEAD trial will read out in about 4 years now, so that's not too far off. And donanemab, I'm not sure when that, it's around the same time, I think. Yes.
So since we're talking about clinical trials, the reason actually in the meeting Marilyn invited me is that we actually, can you hear me now? Yes. So we actually have a study focused on ApoE4 homozygotes with early symptomatic AD. So they're patients at the same stage like lecanemab and donanemab, partly supported by NIA, grateful. So this study completed 325 patients, MMSE 22 to 30. They have imaging, volumetric imaging, plasma biomarkers at every visit, the year-and-a-half study, all the clinical outcomes, MRI, DTI as well. So we're going to have a lot of really interesting data coming out soon where we really profile them as much as we can. We couldn't do PET scans. We tried to get a supplementary grant for tau PET. We didn't get it. But, you know, the interesting thing I want to share is it's doable. When we first started the study 4 years ago, the AD trialist said we can't find homozygotes to do a trial stand alone. You know, it's hard. We did a lot of effort. We screened 6,000 patients. We're able to enroll 325 with the right inclusion criteria. What's fascinating about them, mean age is 68, like you expect. Half of them have hyperlipidemia. The most common comorbidity is hyperlipidemia, 45% on lipid-lowering agents, mostly statins. Very exciting patterns coming out that we'll share once we're able to have the whole data. Still waiting on the plasma biomarkers. But I think, you know, ApoE4 studies can be done, and they really are worth doing stand-up because they have a very unique phenotype.
You'll just ask us to address some of the points.
Yes, I'll bring up the question from Reynaldo Oria, which I guess you can read. But why don't we focus more on synapse neuroplasticity regarding ApoE4 outcomes in animal models? What is the stage of blood-based synaptic biomarkers for predicting AD? I don't know if you want to comment on those aspects. Also have a word.
I think in some of the animal models, people are certainly looking at synapse neuroplasticity regarding affecting ApoE in different ways. But in humans, there is one PET marker that you can look at, a measure of one type of PET marker to look at synapses. But I don't think it's been validated nearly as much as some of the other methods we use to look at outcomes yet. So I don't know how easy that will be. There are measures of there are synaptic markers that are assessed in CSF and blood, or CSF in particular, that are elevated in people as they develop AD pathology, but I'm not sure that's really marking synaptic dysfunction as much as just some damage that's occurring. So it's not so easy to do that in people. Obviously, we can monitor things that aren't a proxy of synaptic loss, like volumetric brain MRI of different brain regions is probably demonstrating when you lose volume, you're probably losing, you know, axons and synapses and dendrites, and that's a surrogate marker for synaptic loss. But plasticity itself, I'm not sure there's great ways in humans to do that other than cognitive, really. Yeah, we know.
Yes, I mean, just to comment. I completely agree. I think it's hard to at least from the point of view of biomarkers standpoint, it's hard to differentiate synaptic dysregulation versus loss of synapses. There are clearly synaptic proteins that people look at in the CSF, and synaptic loss typically results in loss of many or several synaptic proteins in the CSF as the disease progresses, and that's obviously an important part of monitoring synaptic health, in addition to looking at neuronal health with markers such as NFL. There are some approaches that look at synapses also in human brains based on PET tracers for SV2, for instance. Those are becoming a bit more popular, but they also reflect mostly the health of synapses and not so much dysregulation that may be induced as a result of ApoE4 in preclinical models. So that is something that's extensively studied in preclinical models as well as Dave mentioned.
Anastasia, before you ask a question, we probably should move on to the second groups of things that we put out there. So maybe your point has to do with it, but the next step has sort of, I think we should probably talk about what do people think mechanistically about how does ApoE most likely affect AD risk? How does it most likely affect progression? I mean, there's no answer to this yet, but I'm sure people have opinions based on different sets of data. I don't know if you wanted to
I can
talk
start.
about that or not. Or...
I think our naive understanding of what's going on is that ApoE, which is lipidated, ApoE in particular, is providing a shield or co-aggregation which prevents amyloids and tau tangles from being recognized by Glia and clears. And from this kind of mechanistic perspective, just one small comment in terms of timing of treatment. And Alzheimer and late onset Alzheimer disease is disease of aging. It means that people live with 40, 50, 60, 70 years with certain offense. And we know that the amyloids appear many years before clinical manifestation of disease. So from a standpoint, biochemically, how long do you need to treat? And quite possible that because it takes so many years for the disease to develop, and if you remove or clear temporarily the offending tau tangles and plaques, it's the effects of this treatment can work long term. But it remains to be seen.
So, yes, Julia, did you want to?
Julia, TCW from Boston University. So I would like to talk about the ApoE patient case. That what other method other than lowering ApoE we can think about related to the mechanistic study. So I think we try to understand a lot of presenters and speakers talk about this ApoE4 effect in all these different cells, and if these cells going through challenging process of amyloid and tau, and one hypothesis that I think is very interesting is that most of the thing we see is degradation has a problem, and we see that maybe epigenetic is involving in these dealing with these changes of cell state and dealing with amyloid and tau, and if we can identify to controlling those changes, we can think about it as a different way of the therapeutic aspect to modulating epigenetic changes. And the other part that we are interested in drug development is, continue understanding multi-ohm levels to look at what is the downstream target that we can control, whether we identify some small molecules to try on.
So just to restate what you're saying a little bit, Nick, are you saying it might be worth understanding if different ApoE variants lead to epigenetic changes, which then are what leads to either worse outcome or better outcome, something like that? Yeah, I don't think that, to my knowledge, that hasn't really been studied very much. So.
That's why I think it's what else we can do in the future. That's what I would like to share my thoughts.
That sounds like a lot of study to figure that out, though.
But eventually, like a biologic is great for lowering ApoE, but for the long-term and wider way of treating patients, we also have to think about the development of small molecules as well. So that's why I was thinking about what other way we can approach those systems.
I did want to bring up Vivian Chung's eRNA that's part of this R-loop that seems to have, I think we should encourage more work on that because it could possibly be really useful. It regulates ApoE levels. Yeah, right at the promoter. And it definitely is there and it's expressed. And it's an R loop, but when it's expressed, it seems to turn off ApoE. So we need to, I think.
From her talk that it's not from Alzheimer's disease, but as you mentioned, like she actually removed a bunch of kinases, and then one of the most significant genes is fluctuating with ApoE. So that's what I.
She came about it because she's studying juvenile ALS, and one of the defects there eliminates R loops. So ApoE started being expressed everywhere until she followed that up. But I do encourage that. And the other thing would be the Christchurch. We've heard some about that, but to me it seems to be as affecting tau, which is really the clinical aspect. And the other thing mechanistically I throw out is, do we know why amyloid storage leads to tau? And ApoE4 involved in that? I think some people would say we don't understand that. There is evidence, and of course there are model systems that the amyloid induction are stimulating more tau pathology, at least in a system that Virginia Lee developed that we've utilized a lot, suggests that microglia are required for that interaction to occur. So if you don't have microglia, it makes it much worse. If you stimulate certain aspects of microglial function, it makes it better, including the effect of ApoE3 Christchurch on that. So I think there's interesting things to learn about. I mean, that's not the final story. Maybe there's other ways that it's doing that, but I think that's really important because ApoE plays a role in that process, which Christchurch variant appears to be possibly doing. Would suggest, yeah. It's a major pathway to target, whether it's through ApoE or other ways. Right. If tau is in fact the final clinical hammer, so to speak, maybe that's the place to really focus on what people think.
Anyway. Yeah. So I'm not from this field. I've looked at it a bit and talked to some people, and I'm very puzzled. There seem to be 30 or 40 different mechanisms, many of which have been discussed here, and that would be totally unique, as far as I know, in terms of variants we know about and other disease phenotypes. So I wonder if we could maybe someone could explain to me how you think about that. Are those 30 or 40 all really relevant? And if not, what should be done about deciding which ones are relevant? I know that's sort of where you want to go, but to me it's very odd. And just to add to that a little, I think that there is a sort of lack of cohesion in the field, as I see it, in terms of trying to address how do we assemble, how do we integrate this knowledge, how do we assess it?
Yeah, so I think you welcome to our world, first of all. I mean, in a way, it's not surprising to be facing such a daunting complexity in the biology of ApoE. It's a protein that is expressed in multiple cell types, particularly in the CNS. It's a protein that is secreted that can affect any CNS cell type. It has a role in the periphery as well. So that's inherent complexity, and tied to that is also the modeling that we have to sort of achieve to be able to understand the biology. And I would say that the preclinical modeling, at least in cell culture, has been a bit more challenging than we would have hoped
and there are a lot of people who have worked on this for many years, and they know this much better than I do. The preclinical models, on the other hand, have been very, very compelling, and I think a lot of compelling work has been done also in the mouse model. But I think that I think personally we have to understand the effects of the ApoE alleles that are sort of very proximal to ApoE, as opposed to focusing on events that are in the biochemical cascade that are distal. And I think there's a nice review that Dave and Bu and others have published recently, trying to sort of capture the ApoE cascade. I guess very similar to the amyloid cascade, but really sort of custom made for ApoE. Try to sort of provide a hierarchy of mechanisms and events that sort of reflect what happens as a result of the various ApoE isoforms. And that's a very helpful context, in my opinion, trying to get to the bottom of that complex biology. Yeah.
Li Wei Cai from MIT. Actually, I really like your terminology, Gil. So ApoE cascade, ApoE4 cascade, ApoE3 or ApoE2 cascade. But I feel that in order to be able to fully keep track, right, of what's going on, I would like to know how much effort there is to sort of make effort in identifying ApoE, say ApoE4 specific biomarkers, especially when we talk about intervention, right? I mean, right now, I think people pretty much just follow the overall general AD biomarkers. But, I think it will be very important to really start to think about ApoE4 specific biomarkers. That I think, if we have that, I think that would really give us much more power to not just follow the therapeutic intervention progress, but even start to really look at people and predict, right, which ApoE4 carriers are more likely to develop AD in a certain time period.
I think that's kind of what Carlos was getting at in his talk, that there may be some markers that differentiate based on ApoE4 or not. And I don't think he talked about which ones were specific, but I think he has some data on that. But that will need to be validated, whether they're really biomarkers for the E4 state or for E4 with AD or but I think you're right, that would be helpful.
Yeah, and also when we talk about E4, we kind of just refer to just the general population of E4. But even within E4, I think there are a lot of them, it's not just E4, they probably carry other risk alleles as well. So, I think they are not homogeneous. By far, they are not.
Yen? Richard Mayo, Columbia University. I'm trying to, listening to this, I'm trying to get my head around the numbers of people that we might have to be dealing with. If we, for example, there's something estimated to be about 6 million people in the U.S. And so if you do that ApoE4 frequency, that's probably at least half of them probably have an A4, and sort of a smaller number are homozygous. And those are the ones that we know have Alzheimer's disease. And we all know that there are people living with Alzheimer's disease who haven't had a diagnosis yet, maybe seeing their GP and the family doctor saying, don't worry, it's a normal aging or something like that. So I think we have to think about how we're going to scale treatment. I don't know that, all of these early interventions like gene therapy and AV therapy and things like that, those are useful models to sort of figure out how to change things. But then at some point we have to come back to reality and say, well, there's 6 million people. How are we going to get this on scale to treat them? If it's a rare Mendelian disorder, I think these kinds of interventions, SMA and things like that, that works very well because there's not that many patients. But when you're talking about maybe 10 million people in a country like ours, and that's just, and then you think, well, those therapies are not going to be inexpensive. They're going to be very expensive. And that cuts out all the people who are in the lower socioeconomic status. They're not going to be able to get that treatment. So I think we have to think of it in two ways. One is there's going to be a lot of people who have the disease, and you can't prevent it. And maybe you can slow it down, but you can't prevent it. Then there's going to be a whole bunch of other people who are coming up who are going to be at risk. So I think we have to divide this into, let's try to think of what are the therapies we can do to slow the progression, which is amyloid antibodies and things like that. And then I think we have to really give it some thought. What can we do to prevent these younger people from getting into that stage? And that's a bigger group of people. And so maybe we ought to reframe how we address this. And whether it's and ApoE is certainly a good marker in that. And I like Eric Hyman's idea that you look at people who are ApoE carriers and then see, do they have biomarkers as well? Because the biomarkers start to change about 20 years before things happen. So maybe there's a way to use all of these things to our advantage and sort of come up with different strategies. I think we've got a big problem here.
Okay, well, so before going back to the last question, which relates to yours, like what are the dominant mechanisms that ApoE is related to Alzheimer's disease for? And one way to think about this is that as far as I know, you don't start seeing cognitive impairment that's clinically evident in people unless they have the neuropathology of this disease. We know that ApoE affects different aspects of at least amyloid. It affects something about tau. I mean, those seem to me probably important issues to work on in how ApoE is affecting the disease, because you don't see people who are 80 years old that are normal, even if they're E4, unless they also have, or are abnormal, unless they also have AD neuropathology. It's not like they're getting this dementia without getting that pathology. So I think at a very high level, that's ultimately, I think, what we should be using, at least as a marker for what the effects are.
I just say I agree with you, Richard. We really have two different groups. And I saw an estimate of something like 250 million people are actually at risk for Alzheimer's, if you include all the people coming up. And so it really is two different problems, because we're not going to repair what's already, the damage has been done. So we can slow it maybe. But we really, and so relative to the original question of, whether we should go in and try to do therapies on early, I do think that's where the money is for long term. I mean, for really what we want to do. So, maybe we should encourage that more.
I think an interesting case is the semaglutide, explosion, Ozempic and others, which I'm sure everybody's familiar with. I mean, we're talking about incredibly large patient population. This is a large molecule, initially for type 2 diabetes, and now, it's being used for obesity as well. But the point is, we're talking about millions of individuals, and there are now prevention studies as well, trying to understand whether we can actually prevent type 2 diabetes, very similar to what we're thinking about when we think about Alzheimer's disease. So I think that is a case study that will be sort of shaping up the, how we think about Alzheimer's given. That's right. And you made a very good point about it. I think it's a serious.
I personally, at some point, that will have to be a political problem. Yeah, I know, but I think Jen was next.
Yeah, this time it's actually not a question. Comparing to everyone here, probably the youngest one in the field, but I try to, because I have some thoughts about the gentleman's question, how chaotic our community is. But luckily, we have a centered team that we know for sure is the strongest risk factor. In my own opinion, I feel like we are at the time where the tumor cancer field, when they first time see the P53, and then they were thinking that this is the center, expands everything. Maybe we're at the same age. We're just trying to focus on this point and then nail down the downstream and try to look for the upstream and try to understand the process. Potentially, it can be from, for example, Dr. Yaker's study that's involving a very essential cellular process. That maybe they explain everything we saw right now, maybe not. But I think we are at a very important time as the ApoE-related science. We know a lot of new data from GWAS, and we can see from the new biomarkers to see if we can actually find when is the actual critical point when the ApoE4 influence into the disease progress. I think that eventually we can get down there.
Michael. Hi, Michael Haney from UPenn. Just a kind of a follow-up on this question of scale and treatment. I think I totally agree that therapy approaches for the number of people with this disease might benefit from understanding some specific mechanism that can be targeted with other approaches, small molecule approaches. And it seems like what is very little known about is the way the ApoE variants influence the tau pathology. What is that mechanism? How does the work from Chen showing the Christchurch variant lowering tau pathology, the E2 lowering tau pathology? How does that work? I think it's a big kind of spot of under investigation in the field. I just wanted to throw that out.
Great point.
Hi, I'm Priyanka Narayan from NIH Intramural. I just have kind of a potentially controversial, but maybe from my basic cell biology perspective, I think hopefully thought-provoking idea that I've come to the field also with a similar perspective as this gentleman who put us all on notice. But I think, ApoE is not that interesting a protein in the context of protein function. It's not a kinase, it's not a phosphatase, it's a lipid transport protein. And lipids are involved in nearly every process of cell biology in every cell in the entire body. So it is not surprising that what we've stumbled on is incredibly complicated, but may have simple origins. But I think we, maybe unlike other fields, have not looked at that origin, and maybe a thought-provoking idea is maybe lipid defects are the origin of a lot of these phenotypes. Now, how they actually influence pathology and disease progression might lie far downstream of those origins, and maybe these are just kind of cellular handicaps that are imposed by lipid defects that might get exacerbated in times of stress. But I think it's maybe an idea we can think about is there is an origin to a lot of these phenotypes that might lie in more simple mechanisms that manifested very diverse consequences depending on when and where they occur.
Just a counterpoint to that, even though it may be that what you're saying is correct. I mean, most neurodegenerative diseases, including Alzheimer's disease, are disorders of protein aggregation, right? So what leads to protein aggregation in the case of autosomal dominant AD, let's say with a presenilin mutation, is not necessarily related to what the function of presenilin is, which is to cleave notch. It's this gain of function which happens to cause this protein, which normally isn't amyloid, causing amyloid in everybody, which to be more prone to aggregate. So it's related to what that protein does, but it's like not its normal thing really. So it could be in the case of ApoE, for example, that it's simply, unfortunately, it facilitates amyloid aggregation in addition to its normal function as a lipid transport. So I'm just, I don't know what the answer will be, but, or that's possible. Yes, that's certainly possible.
And I have to add that, this is a valid point, and I think that the field has witnessed a lot more effort in that space in the past 3-4 years. There's a lot of genetic evidence pointing toward lipid dysregulation playing a huge role aside from ApoE. There is transcriptomic data. Lee Ray mentioned this data yesterday, pointing toward lipid dysregulation as well, other metabolic pathways as well. So I think it's definitely the sense that it's disease modifying, and the key is to figure out exactly how, aside from the pathologies and whether those dysregulations sort of converge on the pathologies as well, which is also, somewhat likely because that's what defines the disease in the end from a pathological point of view. Right? So
great
Yeah,
points.
and ABCA7 is a strong risk factor in African Americans, so which is obviously another lipid carrier.
Yeah, actually, add on to that point.
Oh, sorry. Go ahead.
Okay, I think, I mean, you know, to that point, I think, you know, previously we sort of all thought that ApoE4 sort of directly aggregated and cleared A beta. I think now that's somewhat in question. Like, I think that's still a question in the field, sort of what is a cascade of events that maybe is starting from endosomal lysosomal trafficking defects, but then is carrying on to other effects and causes A beta accumulation, et cetera, and what is, you know, more direct effects of ApoE and specific cell types. I think some of that still has to be worked out.
I think going back to what Priyanka was saying, probably even more controversial, but maybe even A beta production is something that plays a role in the very same origins that lipid setup. I think I could never wrap my head around this process that evolved to make a neurotoxin that kill aging brains. It must be something that A beta is doing. There is some evidence that maybe actually playing a role in lipid metabolism, actually working as ApoE, as a cholesterol extractor from endolysosomal membranes in general. So yeah, maybe we need to rethink and going back to the you know, origins of what ApoE and maybe A beta does, although we don't really know what A beta does. I think it's been underexplored because it's been mainly taken as a gain of function toxic thing that, but you know, it's made during injury, it's made, I don't know. I always find it a bit difficult to accept that this molecule doesn't do anything other than implementation.
Good point. I'd like to take a quick question from the chat to make sure we pay attention to this. Apologies for not taking all the questions. There's one that I think is a really important question, which is, do we really have to understand the cellular mechanism for the purpose of the therapeutic development? And so I don't know if you have, I have some thoughts, but I don't know if you have some thoughts on that. But.
No. You probably have different thoughts.
I think it would be helpful, but not necessary.
Yeah. I think it goes back to what Francis was saying at the very beginning. I think it's a very valid point. We don't have to figure out everything before we develop therapeutics. That would be a problem. But I think that it depends a little bit on the therapeutic strategy as well. Like there are, like if you think about ApoE lowering strategies with an ASO or some equivalent approach, what you need to care about is whether you have target engagement, namely can you decrease the ApoE levels, and then at least in the case of Alzheimer's, you have a lot of biomarkers you can look into that have been already looked at extensively in clinical trials, like A beta levels, the tau level, etc., NFL. Do you need to understand everything about the consequence of ApoE lowering to move forward into the clinic with this? The answer is no. The more you know, the better it is. I think that one aspect that is personally I think is particularly interesting is understanding whether the pathway biology can inform the biomarkers, because the biomarkers are critical to understand whether the drug works. So now again, you could say, well, we already know we have the right biomarkers. We don't have to worry about the pathway biology. I think in the case of Alzheimer's, we, in a way, lucky to have great biomarkers already now. In a lot of diseases, we don't have great pathway biomarkers, and that's problematic. I think to understand whether any therapeutics actually work. So, or I mean, you can't, but even to establish the dose that you'd like to pursue in late-stage clinical trials, you need to understand beyond target engagement whether you're moving the biology in the right direction. And I think so. In that sense, I think figuring out the pathway biology or the mechanisms can be important and enhance your trials. But yeah, it's not an absolute requirement.
I also saw another couple people have agreed with Julia that there's we should look more on the relationship between epigenetics and ApoE. I'm not sure what epigenetics means in this sense, but certainly we know that a lot of looking at all the diversity, that there's different controls on ApoE expression, and so I do think that getting clear ideas of natural controls of ApoE is important as we move forward. So for me, that's a positive.
Well, I've been struggling for more than 3 decades trying to figure out how we're going to attack a molecule that is part of essential human metabolism. So, you know, we're talking about trying to modulate an effect of a protein in the brain, but it's involved in so many metabolic processes. And the one thing that we have been talking some of the presentations about is we've got this effect. We know it's variable. So we know that there has to be factors, whether they're environmental or innate, that modulate the effect of ApoE4. And there's already work now ongoing, and the question is, should we be looking upstream? What are the factors that influence ApoE? Or downstream? What are the effects of ApoE on something else? And should we be looking there? And I don't think the answer is one, but I just struggle with the idea of tackling a protein that it is part of just natural human biology.
But one way to think about this was brought up a little bit by Joachim, in a sense that if you think about the most impact of any drug over the last 40 years are probably statins. They affect cholesterol biosynthesis, and yet they can be used effectively to suppress cardiovascular disease. So there's examples where you can affect some fundamental process as long as you don't eliminate it.
Well, that's yes, and I actually had this discussion with Francis last night, and that is he was regaling me of all the successes that there have been in other areas, cancer and diabetes and rheumatoid arthritis, and he was just knocking on us. Why haven't we done it in Alzheimer's? And I told him, I said, there's one difference between all those successes. Those are all systemic, not brain. And that's, I'm just expressing my frustrations and my concerns. And I think we need to be thinking creatively.
That's a good point. That's a very good point. I think that you touched upon an important topic around the progress, a lot of progress in witness in systemic diseases. The CNS is unique features, of course, and also the blood-brain barrier, which I think that has been a major hurdle for drug development from the very beginning. But as you know, and my company does a lot of work in that space, but pretty much every company now is thinking about those types of approaches, like increasing, for instance, the delivery of large molecules through transferrin receptor-based technologies, the brain shuttle, whatnot. I think this is helping a lot with the CNS exposure, and I think we'll be presumably in a better position in the next few years to understand whether our drugs can affect the CNS when this CNS exposure is improved. And I don't think it's been a huge problem for small molecules because chemists have known how to make brain-penetrant small molecules for a long, long time. But certainly in the field of large molecules, this has been an issue, and there's a lot of room for improvement. So, the valid point, CNS is a little tougher, as we know, so...
Anastasia.
So I actually have a comment in terms of modulation of fundamental human biology. I understand the concern, but with that said, we have several examples right now. If we are talking about cholesterol metabolism, as David said, with PCSK9 inhibitors, you can bring it extremely down. And if you go to the doctor, they tell you 100 cholesterol is a norm. What is defined as a norm? The norm is defined what average people have. And right now, with inclisiran and very other potent PCSK9 inhibitors, people were able to bring it down to 50 and bring it down to 25. And if you look at the clinical data, the 25 is fantastic. There's almost no cardiovascular event, and it's profound decrease in a stroke. With GLP-1 agonists, absolutely, it's the same thing. And with PCSK9, there is right now programs where you gene editing once and for all. So I understand the argument of fundamental biology, because there is multiple examples where manipulation of fundamental biology allows to expand the lifespan. And from that perspective, ApoE for the brain, as long as you're doing it selectively, might be one of those approaches. And the other comment which I wanted to say, just purely economical. I do understand that drug development is extremely expensive. But in the long term, as soon, like plus 20 years, as soon as all the patents are off, it's all about the drug cost. So the current cost of antisense NSIs per gram, if you do kilogram scale, is a couple of $100 per gram. The expected dose to treat the patient in CNS, let's say it's 100 to 200 milligrams a year. So we are talking about the cost of under $100 to treat a patient. And then we were also talking about the fact that maybe you do not need it all the time, but you need it periodically. So while, yes, it will require a lot of effort, but actually the cost of it and benefits for the medical system, health system, if we reduce the appearance of Alzheimer's, can be enormously beneficial.
Other people have questions. We do want to move on to the next topic, but maybe you could do the questions or comments now, and then we'll move on to the next topic.
This time it's a question, and it may be related to the next section. So it also related to the fundamental biology of this thing. So actually, if we think about the ApoE, this is very unique protein because it's strongly associated with the lipid, right? It's a lipid transporter. So now we are thinking about manipulating the protein levels, removing ApoE and through the cell type specific, whatever. When I was doing my own research, I also get very puzzled by this type of situation. So if the cells do not express ApoE, do not secrete the lipid out with the protein, the lipid does not disappear. It stays in that cell. And with those lipids accumulating, are we thinking that there's no impact in the cells who supposed to donor or deliver the lipid? In this case, in the future, should we consider that part into the research to make sure that this idea is safe and don't have any other potential long-term side effects? I guess that's my kind of question.
I think it shouldn't preclude us from doing the therapies that are
no, I'm not saying
but I think that's something to explore while that happens. That's a good point. I think Allison had it.
Yeah. Thank you, Dave. And I don't remember what the next topic is, so maybe I'm touching on that. But
it's on the ancestry is the next thing.
Oh, okay. So we seem to in the AD field always sort of focus on ApoE4. And I think we had a few talks that were around ApoE2 and Christchurch, but the more general point, I think, is that we should be thinking more about protective variants that we can identify from genetics, because nature's done the experiment for us. And we haven't really had much conversation around that. And to follow up on what Eric said earlier, I think when you look at cohort studies, there are way more E4 homozygotes who are cognitively healthy than people think. And just one example for you, in the ASPRIC study, there are about 12,000 people who are the genotype. They're all over the age of 70, cognitively normal when they were enrolled. 1.5% of the people in that study are E4 homozygotes. In younger people, it's about 2.25, something like that, the frequency of E4 homozygotes. I think that's much less depletion from an aging population than most people would think. And so there must be something, genetics, environment, epigenetics, that's preventing those people from getting disease. And those are the kinds of people I think we should be studying more.
I am, as you know, a big proponent of looking at protective factors. I agree with you. Nature has done the work for us in these. We just have to just figure them out. So I agree that is an area we need to move in. Now, the next topic is concerns Asian ancestry. We know that the risk for ApoE4 is highest in Asian ancestry. Our previous discussions, though, have really focused on European and African ancestry, and now Amerindians, but we need to bring in the Asian information and really begin to understand why Asians have such a much higher risk. One of the things that is, at least from my personal research standpoint, has been is just it's difficult to get samples. There's a few more hurdles that way, and so I think that's something we as a group need to try to overcome. And I don't know if there are people who, there are certainly, perhaps we have the ACAD, which has started, and I guess we have samples coming in now. Tissue is needed, certainly, for studies. I don't know if people have any thoughts on this, but I know Lindsay.
We've put 4,000 whole genomes of Korean half cases, half controls into the ADSP.
Is that K-based?
No.
Okay.
This is the GUARD study.
Oh, there were GUARDs, yeah.
And another comment, Jeff, that you raised, and I'll focus only on the Asian component, but it's true throughout. And now I'm wearing my statistical genetics hat. If you look at the relative risks with E4, they're also aligned with the frequency of E4, which is most common in folks of Black African origin, then European ancestry, and lowest in several East Asian populations. So the effect can be comparable. But once you start tinkering with the frequency,
with the relative risk.
Yes, the odds ratios go up. So in fact, the difference, particularly between the Asians and the East Asians and the Europeans, to a large extent, and I won't, it's not just, but to a large extent can be ascribed to that.
Well, that's an excellent point. We just want, our last paper, we kind of excluded any comments on the Asians, somewhat because of the papers from Korea, but it is a subject that we need to, I think if we can include more data, that's the last kind of diversity that we have, that certainly ADSP is trying to bring in. But,
other than the data that from your group suggesting that in people of African origin, that it may be that they're expressing lower levels of ApoE4, for example, based on your work. Is there, other than what Lindsay presented, is there any evidence that Asian populations express E4 at higher levels?
I'm not aware. We've attempted to get material to look at that, and we haven't been successful, but maybe others have ability to do that. But I think it's an important question, for sure.
Regarding that, I think that's we talked yesterday a lot about the cis elements in the ApoE genomic regions, but the common SNPs in other genes potentially interacting with ApoE. I know Alison talked about that, but several others. I think that there are interesting cases, and I don't know if it has any, if it's driving some of the ancestry-dependent risk, but I can think of one case, for instance, where
an Alzheimer's gene called PLRA, and full disclosure, will work on it, has protective variants, and it seems as though protective variants are only manifesting themselves in the E4 context, in E4 carriers. I assume it's mostly Europeans in that particular study, but it does beg the question of whether modifiers of ApoE4 in an ancestry-dependent fashion can sort of immodulate that risk. And I think it's something that one ought to look at, right? And I think we are doing that.
Isn't the data for that, at least the experimental paradigm, suggests that's an effect through myeloid cells?
Correct, yes.
I just wanted to add, sorry, it's Priyanka Narayan from NIH Intramural Program. One kind of a little plug, but I think a resource that we're generating here at the NIH to kind of enable the cellular studies and molecular studies of iPSC-derived cells in diverse ancestral backgrounds. So we're curating a collection of PBMCs from ADNI that, full disclosure, accounted for every non-European ancestry sample that we could get from ADNI-3, reprogrammed them into iPSCs. That is ongoing and deeply QCing them. These are available through the NCAD website. I just navigated to share with Julia, and I can help anyone else who's interested. There are a few of them that are available already, I think around 48, but they are going to be released on a rolling basis. I think they are not powered enough within this study to really draw broad-reaching conclusions on ancestry, but I think it's a great starting point to improve diversity of disease modeling, because similarly to genomics, I think our iPSCs tend to come from mostly Northern European lines or other very well-represented ancestries rather than underrepresented. The plug for that resource.
Yeah, we also have a lot of different ancestries as well. I think the real question is tissue, which is what David was referring to. We really need, autopsies are not as common in many areas of the world, and that's one of our problems. Not problems, but hurdles.
Dr. Marcora Manzana, brains are not easy to get, but blood is. Is there any evidence comparing, say, Caucasian to Asian that ApoE levels track genotype in plasma? And what about cardiovascular risk? Is the effect of ApoE4 also more penetrant in Asia? I should know this. I'm sure I read it, but I don't remember. Is it more penetrant in Asian compared to Caucasian? And
you mean in terms of cardiovascular risk? Is that what you're asking?
Yeah.
I don't know if anybody knows that in the audience, but
or plasma lipids, plasma lipids and cardiovascular risk. I mean, there are huge GWASs on this, so I couldn't Google it now, but I thought if you guys knew.
Julia has another.
Julia, TCW from Boston University. Thanks, Priyanka. I would like to also advertise about there's other ethnic group study of ApoE dependent manner. We created an ITSL from U19 so it also have the genetic data as well as a lot of people are from WashU and Mayo. They have other clinical information. So there's a website as well. You can actually reach out to us. Me and Sylvans is having 4E, and we are distributing ITS as well. So I would like to share the source as well.
Oh, thanks. That's a good point to bring up. There was, we moved to the next thing, but I just had with a comment regarding ApoE and its role in lipid metabolism. How might that be linked with? We really didn't. Even if the effect of ApoE somehow is on something related to protein aggregation or ultimately something else, there is a lot of data showing that if you increase the main lipid, the main protein that's in the brain, but also in the periphery that lipidates ApoE in the brain, it's ABCA1. And if you increase that function, it actually suppresses a lot of the effects of Alzheimer's disease pathology, at least in animal models. ABCA1 also changes in it that make it a risk factor for AD clinically. So I think that's another indirect way of targeting a form of ApoE that probably is going to lead to protection if you can modify that function of that protein.
I think ABCA7 also is involved in lipidation of ApoE.
It doesn't look like that, actually.
Really?
Yeah, it does something, but it's not, I don't think that's the main effect.
I'd like to bring up a question from the chat. It has not come up at this workshop. There's an anonymous person providing an sort of incentive to look at the studies of the microbiome in relation to the ApoE genotype and AD risk. I know it's been studied more, perhaps in the context of Parkinson's. Dave, you have some experience with that. Maybe you want to take this one.
We've done a study in animal models showing that when you manipulate the microbiome in different ways, you can be neuroprotective in models, for example, of tauopathy, and the effect is different based on ApoE genotypes. We see a greater effect in the presence of E3, for example, than E4. Whether or not, I don't know how that will give us insight into the mechanism, but I think it's probably related to an effect on inflammation, systemically, and maybe affecting the brain that then differs based on ApoE genotype.
I was thinking that, I mean, it could be an impact, could have a huge impact in terms of the ancestries, right? Different diets but.
Just a quick comment about the microbiome and Alzheimer's disease. I think Dave's paper is beautiful. The human data is, I think, almost sort of intractably compromised by the fact that most Alzheimer's patients are on donepezil, which speeds up the gut. So I'd be very leery about that.
There is, just to counter that, there is a recent paper that came out in Science Translational Medicine, happens to be from other investigators at WashU, that looked at people who are cognitively normal, who have preclinical AD based on biomarkers or not. And even there, they see differences in the microbiome constituents between the groups on average, not in any one. So maybe there's something to that.
No, I think it'll get better. I think it makes more sense in PD. There's a much tighter, connection to the gut. It could have a role in AD. I would just be wary of papers prior to the one that Dave just mentioned, because donepezil is going to drive a lot of that change.
Inflammation probably through input, I would guess.
You'll probably move on to CAA, and because that is an important current issue that a lot of people are dealing with practically. So some of the things that we laid out were what are the impact of E4 and anti-beta immunotherapies, effects versus the effects that one sees versus ARIA, can this be avoided, improved on. Maybe just to comment on this, it's as you probably all know, it really looks like the amyloid-related imaging abnormalities that occur with anti-amyloid immunotherapies really, they occur in people who it's due to the CAA being there and somehow the antibodies reacting with it. Maybe because they can't get rid of the material there, they probably are inducing an inflammatory response from perivascular macrophages. It's not entirely clear, but it really is a CAA-related phenomenon that the antibodies are affecting. So one issue would be, can this be avoided with future therapies like other treatments that affect amyloid or CAA? And what's known now is that in very early studies in humans, it looks like one of the antibodies that Roche is developing, if they get it to cross the blood-brain barrier to a much greater extent, it might be causing, it removes amyloid very rapidly, and it seems early on it's caused much less ARIA. Now that needs to be validated in larger studies, so that's one thing. And then, I don't know why, but I mentioned this in my talk in the beginning, that we've identified an antibody that binds to a form of ApoE that's only in amyloid deposits, including CAA, and it seems, at least in animal systems, to remove A-beta, including some CAA, without causing ARIA, at least in the mouse. So those are two things that are currently known, but I'm sure there's a lot of other possibilities for developing things that get rid of A-beta without causing ARIA. I don't know what other people think. Obviously, this needs testing, but it's certainly clinically a huge issue in patients. I don't know what your experience of people here clinically is, but at WashU, we've already treated over 200 people with lecanemab in the last year, less than a year. And I would say that the effects that have been described in the clinical phase 3 studies that were published, we find almost identical data. We don't see it's worse than that. We don't see it's better. In other words, we see with that particular antibody about 15, 20% of people are getting asymptomatic ARIA. We've seen maybe 1% or so getting some symptoms, usually resolvable with either stopping the medicine or giving steroids. But that's what we've seen clinically. Now, donanemab probably is going to cause a lot more or higher percent of ARIA. But that's where the clinical state of things are at the moment.
I would add that it's an issue that goes beyond anti-beta, I would say traditional anti-beta immunotherapies. Like if you think about the Alector trials with agonist anti-TREM2 antibodies, they had to exclude the E4 carriers because there was a high risk of ARIA. So it is an issue that we have to deal with as a field.
That's where it tells you it's something. The side effect is something with affecting myeloid cell reaction. That's it. To CAA.
Exactly. Not directly related to this topic, I think. With another.
We can certainly talk about the second point we raised before, which is the effects of ApoE variants and lowering ApoE4 and risk for other diseases, macular degeneration, Lewy body dementia. And we've talked about this a little bit, that maybe it looks like the risk of macular degeneration appears a little bit lower with E4, a little perhaps higher with E2. Lewy body dementia, E4 looks in several studies to be, E4 is a risk factor for Lewy body dementia. So that might be another disease that one could consider for targeting ApoE4, actually, given the data that's been published from humans. Eric, did you have a...
Yeah, Eric Reiman Banner. I just wanted to point out, if we had a primary prevention therapy for Alzheimer's disease, there's fairly good reason to think that the relationship between amyloid plaques and alpha-synuclein pathology, and the second most common form of memory and thinking problems, is primarily amyloid. So the same primary prevention therapy potentially dramatically reduce the clinical.
No, I think you're right. I think it's about 60% of people who have Lewy body dementia, in addition to having synuclein pathology, have amyloid pathology as well. You're right. That would probably, if the synuclein's downstream in those cases of A beta, then that probably would take care of that. You're right.
On this topic, I apologize for not remembering the authors, but there was an interesting molecular generation paper indicating that the risk for progressive supranuclear palsy, PSP, which is the primary encephalopathy, is inverse for the ApoE genotypes, with E2 increasing the risk and E4 decreasing the risk. With odds ratio, that seemed to be meaningful to me, and with power, that seemed to be meaningful.
I think some authors of that are in the audience. I don't know if they wanted to comment, but...
Yes, thank you. Sorry.
Jerry, when you were mentioning that to us before the paper was out, I had wondered whether, is it possible that you see that relationship simply because so many people that are E4 positive are developing AD pathology relatively early in their life, that they probably then wouldn't get a disease like PSP, which is a completely different pathology, and maybe that's why you see some slightly higher quote unquote risk in E2, or I'm sorry, yeah, with E2, but I don't know if that, that probably wasn't addressed in this unless you had done biomarkers on people. But.
Hi, Mike Rachis from Stanford. Not a huge point, I don't think, but this question about ApoE and Lewy body is very complicated by the dual pathology for sure. We looked in a big series of autopsy data to look at, pure Lewy body hard to find minority people. And in that setting, when you just look at Lewy body, the absence of any amyloid or tau pathology is no effective. It's a relatively small sample because it's autopsy based, and it's the people that only had Lewy body. But in that smaller setting, there was no equal effect on Lewy body. So
there's another paper that did the same thing that you're saying that does suggest it was still a risk factor, which is pure synucleinopathy.
Yeah.
Is there something wrong with that paper?
That was a smaller study. Ours is an extension, yeah. No, that was Tom Montine at Stanford, so we felt a little reluctant to pile on, but he contributed all those data anyway. So but I don't think there's much of an effect of that.
So one issue in that disease is do you think there's, even if amyloid is also involved in many cases of Lewy body disease, is there a reason why one wouldn't still want to take a similar approach of, for example, lowering ApoE4 with that or not? Or do you think we're just not at the point we're ready to really deal with treating that yet because of other reasons? I think clinically you can certainly especially if you use biomarkers, you would know somebody with a syndrome of the early Lewy body dementia and amyloid, and there's no reason you couldn't do a trial with that.
And how strong is the, and I don't really know the answer, I wouldn't ask the question. How strong is the connection with TDP-43 pathology in terms of in terms of the ApoE genotype generally driving that? I know there's a subset of AD cases with region-specific TDP-43 accumulation, but it's not really, I mean, 30, 40, 50% of the patients, it's not completely.
Especially as they're older than 75 or so. Yeah. But the problem is clinically you can't identify who those people are.
So I was just wondering, as a group, do we have any consensus ideas about, is it time to move ahead with therapy promote therapy in humans? We need more models, mouse models. We not taking away from your data, but yeah. But and is dropping the I mean, we have a lot of different possible interactions that could alleviate some parts of Alzheimer's, but is lowering the expression, the one that is the most useful. Certainly, in terms of ancestry, that looks like what nature has done. But I think it would be nice if we had some thoughts on where to go. If we would like to do another paper like the previous one. So I think it would be nice to have some sort of thoughts on this.
Maybe some people could comment that are working on gene therapy of whether it's increasing expression of a variant or decreasing it. But it seems to me while further studies are being done in animals themselves, it seems like trials should start as soon as possible with safe ways of lowering ApoE4 in ApoE4 carriers, or ApoE and ApoE4 carriers. Because there's just so much data supporting that you want to lower that molecule. But I think for the other variants, I think Ron, I don't know if Ron's still here, I think he left, but he talked about data in the animal systems that I don't think it's published yet, showing that if you overexpress, for example, the E2 with Christchurch variant on it, that you're seeing a very large decrease in tau pathology. I mean, if that's correct, that seems like it's really important and something that one might want, assuming you can do the gene therapy safely, you want to get going. But I don't know if you want to comment on that or.
I just would like to comment on ApoE lowering strategies. I absolutely do believe that it's a high priority to go with ApoE lowering in a context of ApoE4 homozygous patients. With that said, we need drastically a white paper from this community to summarizing the genetic understanding of safety for this partial ApoE lowering, because pharma community in general is being very resistant to an ideal manipulation of this fundamental process.
to answer your question, in terms of Christchurch, I think next step is humans, the obvious thing to do. But what impressed me was overall of the conference, and what you started off the conference with in terms of E4 as a toxic gain of function molecule, seems to me that optimal gene therapy would be to elevate E2 and maybe E2 Christchurch, but also try to suppress E4. I think we ought to be thinking seriously of some sort of dual strategy, doing both, raising E2, suppressing E4.
And even in animal models, I've not seen anything published where somebody's done that.
Right.
So.
In regards to the animal models, I mean, since most of the research is done by crossing ApoE4, for example, to ADAD, autosomal dominant AD models, is there anything being done, maybe crossing ApoE4 with, TREM2 mutant mice, maybe with humanized APP, anything that doesn't need ADAD background to study what ApoE is doing in the context of late onset AD? Probably the population here.
Well, one just in some unpublished data, some of the things that we did was just use one of the APP knock-in models it has mutations in order, so otherwise you don't even get amyloid pathology at all. But when you do that, and we cross it onto the E2, 3, 4 mice or ApoE knockout, we get exactly the same result as if we do it with a markedly overexpressing model. So
I'm talking about without mutation, but maybe diet, TREM2 mutation, something like that.
I mean, no one's shown, to my knowledge, convincing data with wild-type human APP in a mouse that you ever get any pathology. So there's nothing really to look at other than. I think people are trying that. I've never seen anybody show a clear cut that you develop pathology.
I think, Anastasia, the original, our previous paper that was just published, really, that was the whole purpose, was to really to show that the data overwhelmingly shows it's toxic and that we need to lower it. Is there, what more in your mind?
Sorry, what do you mean toxic?
It is a negative gain of function. In other words, there's always been a discussion of whether it's not efficient enough or whether it's causing a problem by itself.
No, I think I'm a strong believer, and I understand it's not everybody in the community, is that ApoE4 is a strong gain of function mutation.
Yeah, and that was what the conclusion was of the original working group that we published in the Journal of Annals, Annals of Neurology, sorry, and just recently came out. But so.
I think Big Pharma is not there yet. You probably can hear in this community is that maybe not 100%, but majority believe that it's a gain of function. If you go outside of ApoE community and talk to neurologists in Eli Lilly, Biogen, Roche, their perspective is very, very different. The message is, it's unclear, it may be gain of function, or maybe it's loss of function, or maybe a combination of both. It's fundamental biological process. We have to be super careful. It can have unpredicted consequences. The data is not strong enough to justify it moving it into clinic. I have heard it all with different reiterations. And I'm not an Alzheimer person, so I cannot say maybe now I am a little bit. But we need as a community to have a very strong unifying message.
I've heard this too, and it's, as we've discussed, it's very pleiotropic. It has many, and in some situations, it well may not be efficient enough, and others and that's to be expected. And the ones I've heard of people arguing is very specific situations. But I think overwhelmingly, it was a consensus of the group that the data is overwhelming and that it is a toxic gain of function.
I think that to add to this conversation, what matters, in my opinion, is to have a very clear therapeutic hypothesis
In this case, the toxic gain of function hypothesis is very strong, and there's a therapeutic strategy one can use to test it in the clinic. And I think that to answer the initial question, we definitely have to go into the clinic and test it, because otherwise we'll never have any answers. And we need to, of course, have ways to get into the clinic, and it's not always easy. And a lot of you have ideas on what to do, and it's so you need to be able to get to that point. And sometimes it's small biotechs doing that and getting maybe some clinical data, and then maybe becoming more attractive to the large pharma. But it's a bit of a hurdle, but I think at the same time we have to insist.
Well, maybe NIH can help with this. Ron has taken steps forward, but I agree with you. I think it's time. We can only tread water for so long.
I think if NIH, because this threshold of concerns is so high, and when we're talking about pharma, it's not only pharma. Pharma sits on the board of biotechs, and they control key opinion leaders. So it's not pharma. Biotech doesn't exist in separation. Those trials are absolutely feasible. It's not a question whether if they can be done, it's when they can be done. It's a question of a little bit of money, which in a context of amount of investment in Alzheimer's field, it's honestly neglectable. But if NIH can fund a trial to test a hypothesis on safety and let's say biomarker just in some of the ARIS cohorts and saying, okay, amyloid goes down, NFL goes down, maybe some other biomarkers goes down. And just to generate this data, this will profoundly reduce the threshold to ensure that for-profit money comes into play and do it right.
Anastasia, I agree with many of the comments you made. I mean, just personally, I've probably talked to large pharma and small pharma about lowering ApoE4 for 10 years.
And how is it
like
success?
Yeah, so I think everything you said was correct. They've been very reluctant to even whether it's lowering, whether it's ASOs, siRNA, whatever, until very recently. And I do sense that there's a change now, and I do think it looks like, I mean, I'm speculating that there will be some trials starting within the next 1 to 2 years where that happens. But I think up until now, they've been very reluctant. Is there any other things people wanted to bring up? If so, please get up and ask. Are there any other questions up here that you want to start? Yeah. No. Well, I'm not sure what was left to wrap up, but I think that, certainly from the genetic aspect, we see a lot of different risks, and I think we can utilize that to understand controlling ApoE4 maybe through natural protective mechanisms, as Allison suggested. And we're looking forward to increasing the diversity and getting everyone involved. I do think that well, I'm not a modeler. I do think it's time that we start moving ahead. And as probably one of the older people in this building, I definitely feel it's time to move ahead. I think having not studied ApoE as long as every person in the room, but studied it for a long time, it seems like, given how important this is, it literally is the most important genetic risk factor for one of the most impactful diseases in human beings, and for not have started something in humans yet as a trial, other than what Ron talked about with E2, I think it's shame on us as a whole community of people, not us necessarily as scientists, but us as and I think it's about time that we're finally going to try to do something in humans. But also, having said that, because
because this is a difficult protein to study, there does still need to be a lot more work on understanding its effects. And I think that came out in this meeting. There's really interesting things that ApoE does that I think understanding more will lead not just to the current approach that will probably occur next, but also to new future approaches.
And I suspect that it will end up not being one approach, but multiple approaches that are utilized. Because, you know, it is an important molecule, and it has so many effects.
Eric, if you want to say.
Could I thank our organizers, our NIH colleagues, and in particular Marilyn Miller for arranging this symposium and giving us a chance to interact with other established and emerging leaders in the field.
And Michael contributed quite a bit as well, I believe.
Well, otherwise, it's been a great 2 days, and hopefully we'll stay together as a community and bring new people into the field and keep moving this forward. So.
Yeah, and I think we'll probably be in touch towards some sort of paper or white paper statement with the group. So stay tuned.
Thank you all so much.